I am going to begin the topic that is quality management system. As we all know that whenever we go to the market, we always try to buy the quality products, and that too in a cheaper price. So that too applies in our pharmaceutical products, whether in a textile industry, whether in a medicinal industries. So wherever in a routine system, we can say that quality management system applies in our day-to-day routine life. So, although it is a very vast and widely debated subject among the manufacturers and the auditors, I will try to summarize all these within the very short session and share the best insights I have gained over the past 20 years of my journey in the pharma industry as well as the medical device industry. So, starting with the disclaimer that the presentation prepared is totally based on my knowledge and the perception regarding the quality management system I have learned over the past 20 years of my journey in this industry. Here is the outline, what I am going to explain, what I am going to share with you all people. The very first thing, everybody uh, think about that. What is QMS? What is quality management system? And what are the requirements that everybody should know about the quality of each and everything? So, as being a professional, you all are aware of the quality management system. Either you are directly involved in that processes or indirectly involved in that. So here I am going to just touch upon some of the topics so that it could be covered in a defined time frame. Then secondly, we are going to discuss about the elements of quality management system. Then quality policies, quality manuals, work instructions, records and forms, benefits of QMS. Okay, if we are going to apply all the systems, regulations being a part of this quality management system, then what are the benefits we get from by following these systems to our organization? Then again, it's a part of QMS. Without quality risk management, none of the quality management system is followed. So I have taken some of the topics belonging to quality risk management. How the risk assessment is assessed. So risk assessment methodology, risk classification. On the basis of risk identified, we will classify the risk based on the catastrophic, moderate, low or high risk. So what is quality? As we all talk about the quality, quality, quality. So starting here is the what does it mean when we are going to say that the product that we are going to purchase, that the product we are going to place in the market is a quality product? It means it is made by following the norms and regulations being given by the regulators, by the international and domestic regulators, regulatory agencies like WHO, CDSCO, Pharmacopoeia commissions. So let's see what the exactly quality means. There are some pharma gurus which are also called as the quality gurus. So they have defined the quality in terms of definitions. The very first pharma guru who has identified the quality management system, or we say the quality, is the Dr. Edward Deming. What he says? He says quality doesn't mean achieving the perfection. It doesn't mean you have 100% perfect quality within the product, but it means the efficient production of quality that the market expects. And as per the customer requirements, fulfilling the regulatory requirements. If anybody in the manufacturing of this pharma or the medicines, whether it's a Unani, herbal, Ayurvedic, so you have to fulfill the customer satisfaction. If you place the products in the market that are fulfilling the customer's requirement and the regulatory requirement, then it is to be said as the quality products. Another pharma guru that has identified the quality is Dr. Joseph M. Juran. What he says a very short statement in a 3 words, that is fitness for use, or we may say FFU. So, what FFU means? FFU is very often used in QMS and regulatory context. It indicates that a product or equipment is ready for use. Immediately when we go to the market, when we go to the uh, doctor prescribes a medicine, and he says this, you take the medicines thrice in a day, twice in a day. It means it is immediately ready for use. So he defined the concept quality as FFU form. FFU means I want this interactive session. What I feel, I'm just saying something and you are not able to understand what I'm saying. So it's a kind request. Please, everyone is having the mic. So just say either yes or no. You are getting my point or no. Right?
Yes.
Okay. So what we have said that fitness for use, FFU. This data also ensures that all the required testing, validations, specifications have been completed before the product have been launched in the market, and it helps to minimize the compliance, to maintain the compliances, minimize the complaints, and ensure the safety and effectiveness. And in totality, we can say that our customer or our patient is safe to consume while consuming these the products or while consuming these drugs. Then again, one of the pharma guru, that is the Phillips Crosby. What Phillips Crosby said, he said only conformance to the requirement. So here we are seeing the three different definitions of quality with respect to the customer satisfaction, with respect to regulatory perspective, that he simply says conformance to the requirement. So what does it mean? It means the product is meeting the specific standard, regulations, or the specification as per the predefined specification being prepared by the quality people before the product is launched in the market. It is all about the ensuring of the product, services, or the process aligned with the defined needs, expectations, and the rules. So simply we can say that nothing is 100% perfect. But if you are going to launch the product, if you are going to place the product in the market that complies the customer, that complies the regulatory requirement, that satisfies the customer requirement, then it is being said that it is a quality product. Sorry. Hmm? Yeah, definitely. All these things are being, these are the first scientists who have defined the quality. And later on, there are several scientists in India also who has given different definitions of quality. But it's a long list. That's why I have not included here. I have included the very first scientists that have been defined the quality in different ways. So, QMS for different industries. The very first is ISO, International Organization for Standardization, that is 9001 and 2015. This Indian Standard Organization also defines the rules and definitions for the our Ayurveda, Siddha, Unani medicines, apart from the other industries that being laid down in the market. So what does it says? It says a very general QMS applicable across the various industries, focusing only on the quality management system principles.
Madam, is that ISO number which have been specified over here? Is there
Yeah,
any specific meaning with that number or what it signifies?
definitely ISO specific number 9001 specifically define quality management system for different industries, and 2015 is the year whenever it is being launched first, very first when it was being registered. So this defines the number, sorry, year.
Okay.
Specifically number is fixed. Another one is ISO 14001. So here the picture is in green color. What it signifies in
Environmental aspects.
environmental aspect? Very good. So it is an international standard for environment management system. What the regulator says, if you are going to manufacture any product, that should not contaminate any environment, and for that you have to comply all the norms and regulations being laid down in ISO 14001. So the regulatory agencies define ISO 14001 specifically for environment management system, which proves that the products being manufactured or being placed in the market does not give any impact either in the aerial living beings or the human beings or the soils or the water. So in all the three phases, it shows that your product doesn't harm to all these systems. Then ISO 27001. As we all know that um, we are in a AI system. We are transforming our system in an AI system. So definitely there should be one cyber security in our day-to-day life. We come to know that this has been happened just only because of the cybercrime. So there is a system, ISO defined a system 27001 that ensures that your all the data, all the sensitive data that should not be uh, tracking by another organization, by the hackers, or and the efficacy and the integrity of your document should be maintained throughout the life cycle of the product.
But I mean, ISO 14,001, you had not mentioned the year. I think 2016 is the year.
Yeah,
And now expected that 2026 it is updated version coming
yeah.
soon.
14,001 has been updated in 2020. Recent update is in 2020, but still it is not in rule. So that's why I have not mentioned here the rule because it is in draft. Once it is being finalized, it comes in the year.
What is the right way to represent the ISO with the year or without the year?
Both are applicable. Both are applicable. If year is not being written, it means it is not yet being finalized.
Older one,
Recent,
previous one, we should
recent.
be mentioned. No?
Yeah,
Then we
previous
should
one we
mention.
can mention, but once it is being drafted and once it is under the regulatory agency, then we it's a routine practice. We don't write the years.
But in 9001 and the 13847, you should mention the.
Yeah, 13485 is the recent version of this space. Okay, I will come on the for the medical devices because initially medical devices also comes under the ISO 9001 2015. Later on, some regulators have compiled, come together, they sit together and drafted a different guideline for the uh, medical devices. That is ISO 13485 that comes in existence very first time in 2016, and later on several amendments have been come, but it's still not in finalized state. That's why very first time when it is being implemented in this era, it is being written here. Again, the elements of quality management system. So these are the labels, different levels of document that we have to define under the elements of quality management system. There are different levels, level 1, level 2, level 3, level 4, and level 5, depending on the importance of the document, depending upon the requirement of the document, it is being defined. And these levels are not as standardized. It will be defined as per the requirement, as per the industry norms, as per the regulatory requirement. So if we say that the very first document structure of this quality management system under level 1 is a quality policy. Every organization is having a policy. So what does it mean? It means we prioritize the quality by ensuring the product services meeting the customer and regulatory requirement. So whenever any organization comes in existence, the very first thing he do is he prepare the quality policy under which he mentioned what are the quality aspects, what are the regulatory requirements, and what we are going to implement with this policy. Another one, the quality manual. Quality manual, it typically outlines the objectives and the procedures. What is the objective of preparing this document? What are the objective of this system? And some procedures should be there. So these procedures comes under the scope of quality system procedure, standard operating procedure, standard testing procedures. So all these comes under the quality manual. It should be in line with the requirement of the regulatory agencies. And what it makes, it helps to ensure the consistency, quality, compliances within the regulatory standards. Then moving towards the level 3, that is quality procedures. As I said earlier, that quality procedures in the form of standard operating procedure, quality system procedures, standard testing procedure, standard specifications, standard manuals. So all these comes under the title quality procedures. So under these quality procedures, work instructions and SOPs are come are in a frame line. So what are the work instructions? In every organization, we have 2 3 categories of employee. Someone is leading the department, then someone is assisting that leader, and then someone is assisting that second line person, someone is assisting that first line person. So work instructions are for those persons that are not technically sound, that are they don't know how to follow the system, but yes, likely or unlikely they are a part of system. They are also responsible for the quality, safety and efficacy of the products. So for those employees, those are illiterate, like the washerman, like the cafeterias, like the person who are responsible for mopping and sweeping of all these things, for those people we make the work instructions. So work instruction, there is a guideline. Work instruction should be in bilingual state. One is the local language and another one is the English. Local language is compulsory because if we are saying that that person is not technical, then it is not possible to read all these work instructions in English. So this is a mandatory requirement by the regulators that work instruction should be in bilingual languages. Then formats, forms and records. What are these things? These are the these are the records. These, we may say that these are the retrospective data for the future purpose. If suppose a drug is having a shelf life of 3 years, suppose we are going to manufacture any drug, starting from its manufacturing is having November 25 and its expiry is October 28, then today we know what are the processes, what are the actions, what are the chemists, what are the procedures we have involved in manufacturing or testing this drug. But after some time, after few years, after 2 years, suppose we got a market complaint for that. For investigation of that market complaint, we need to see the retrospective data. So these formats, forms, and records are useful for tracking the data to fulfill the requirement of this investigation system. Okay.
I have some question. In quality manual, how many times we should amend? कितनी बार हम उसका रिवीजन कर सकते हैं साल में?
It's not a mandatory condition. See, if you for this drafting this quality manual, you have to draft a system how to draft a quality manual. What are the contents? If you fix a frame line that within 3 years we will revise the quality manual, but there is an exceptional, this you have to mention one single line also. It would, however, it is fixed for 3 years, but it can be revised at any time as per the regulatory requirements. If there is any updates, if there is any changes in your organization, which directly or indirectly impacts the quality, safety and efficacy of the products.
साल में कितनी बार रिवाइज कर सकते हैं? 2 बार या 1 बार? कोई राइडर है उसके लिए?
No, कोई गाइडेंस नहीं है.
नहीं राइडर,
नो
कोई
नो,
चेक पॉइंट
कोई
है कि
कोई
but
राइडर्स नहीं है.
times we
मैं
should
ये कहना चाह रही हूँ, क्वालिटी मैनुअल को आपको एक डॉक्यूमेंट फ्रेम करना है. हेडर फुटर आपने फिक्स किया, दिस इज फिक्स फॉर 3 years, बट देयर शुड बी वन स्टेटमेंट, इट शुड बी रिवाइज्ड each and every time whenever it requires regulatory requirement. आपका routine review है, sometimes during the review you saw that these points are missing, so just giving a revision, you simply revise the document, giving a revision number, issue number, revision number, issue number, revision number. So within, it is not mandatory to revise any document within 1 year, 2 year or 3 year. But yes, there should be some guideline for which time you have to fix or you have to frame these drafting documents. It depends on the requirement.
नहीं कितनी बार एएल निकल सकता है? मैं ये पूछना चाह रही हूँ.
कितनी बार भी नहीं, वो ऐसा कहीं भी डिफाइन नहीं है. It depends on the requirement.
इन माय नॉलेज मेरे ख्याल से 2 टाइम से ज्यादा हम
ना.
लोग रिवाइज नहीं कर सकते
ना, 2 times का नहीं. मैं इसीलिए कह रही हूँ, एक
पर
स्टैंडर्ड
एनम.
ऑपरेटिंग प्रोसीजर बनानी होती है आपको. उस प्रोसीजर के अकॉर्डिंग होता है. आपको पहले तो आपको फ्रेम करना होता है कि आपको स्पेसिफिक कितना है, बट देयर इज नॉट अ सिंगल गाइडलाइन टू रिवाइज एनी डॉक्यूमेंट twice, thrice or 4 times in a year. You can revise a document 5 times in a year also. You can revise the document once in a year also, but it totally depends on your requirement. Okay. ISO 13485 का क्लॉज नंबर 4 है. क्लॉज नंबर 4 में पॉइंट नंबर 4.2.2 that defines the documentation system, that defines the each and every outline for this documentation, that every organization should have a system to draft and design the document based on the requirement of the or requirement of the organization, based on the requirement of regulatory requirement, based on the organizations. Then benefits of QMS. What are the benefits? The very first benefit we recognize is the simplifies the organization structure. It simplifies the organization structure by clarifying the roles and responsibilities and streamlining the process for the improved efficiency. This leads to enhance the productivity, reduce errors, and better decision making. So, when we simplify the organization structure, there should be one accountable person, there should be some responsible person, and there should be some assistant person that are responsible for drafting the documents. So, by defining roles and responsibilities of each and every person, we can simplify the organization structure. It improves the work culture by continuous improvement, accountability, and teamwork. Of course, it's not a one man show to draft a document, but it's a teamwork. Each and every person is equally responsible for drafting the document, for carrying out all these norms and regulations as per the regulatory requirement. That directly or indirectly impacts the product quality, safety, and efficacy. Another one reduces cost and waste. If you are perfectly following the quality management system during the complete life cycle of the product, then definitely it reduces the cost and waste being generated during and after the manufacturing activity. Then improves the compliances. Whenever the regulatory agencies visits your organization, there must be some ANSYS, there must be some non-conformances. They should be addressed properly. If you are completely following the quality management system, then definitely it improves the compliances. Ultimately, the image of the company is going to be very good among the regulators, among the market, and it improves the revenue of the company. The same thing improves the productivity, then motivates employee. Then it is also a part of quality management system. If you are motivating your employee, if you are motivating your juniors, then definitely they work with a enthusiasm, they work with a positive environments. And if a positive environment creates among the employees, then definitely that ultimately results in the quality product. The last one, the consistent products. Quality is not a one overnight effect of this manufacturing process. It's a consistent product. If you are going to manufacture 10 batches of the same product, for example, we are going to manufacture the paracetamol, batch number 1, 2, 3, 4, 5, 6, up to 10. Then each batch is having the consistency in terms of quality, qualitatively and quantitatively. It should be consistent. Then increases the customer satisfaction. Whenever we purchase the medicine from the market, everyone has the focus on the uh, impact of this medicine. Suppose it's a very good image of सिप्ला, नहीं हमें तो अगर जैसे भी कोरोना में डोलो सबसे ज्यादा बिका है. डोलो में क्या था? ओनली पैरासिटामोल 500 mg, बट द ब्रांड नेम ऑफ डोलो इज vastly distributed among the people because it is a brand products of that particular organization. I'm not going to say the name of the organization, but yes, the name Dolo is very vastly known by the organizations. Another one, it supports the continuous improvement as per ICH-Q10. ICH-Q10 is International Conference on Harmonization, which defines the quality guidelines for the products being launched in the market. So, in that quality guideline ICHQ10, it also supports the continuous improvement. It doesn't mean quality is a one time product, one time statement, but yes, continuous improvement should be there. Whether in terms of documentary improvement, whether in terms of manufacturing, whether in terms of testing, whether in terms of in removing the old, replacing the old technology with respect to the new one. Then streamlining the training and competency management. So whenever any person joins the organization, he is not well trained. Although he is having the academic qualification, although he is having some technical experience, but it belongs to the previous organization, it belongs to the previous academia. But whenever someone joins your organization, definitely you must have a system to train that particular person. And that training is called as the on-job training. On-job training, whether it's a top one management, top two management, top three management, but everyone should have to be aware of the system, policy, manuals, procedures, along with the uh, pros and cons of the not following the systems. Then enable deviation, kappa change control and complaint handling. The everyone belongs to the part of quality should know about how to manage the deviation, how to draft a corrective and preventive action, how to draft a change control, and how to handle the complaints. Then it is used to ensure the quality, safety, compliances. Obviously, it also builds a culture of quality and prevents failure before they reach the customer or the patients. So, by reviewing
Question है
all these things, we can say that if we follow the particular quality management system in our organization, then definitely the organization is being benefited by these points.
मैडम अगर सीरियल नंबर में मतलब मिस्टेक हो गया है इंक्रीमेंट नंबर में तो हमें कापा भरना है कि नहीं भरना है
नहीं,
तो उसके लिए
सिंपल
क्या
एक इंसिडेंट, सिंपल एक इंसिडेंट आपको करना है.
उसके लिए क्या फॉर्मेट रहता है
इंसिडेंट,
मतलब उसमें
इंसिडेंट,
मतलब क्या टू टैब्स रहती हैं उसकी क्या टैब्स होती है मतलब क्या उसका हेडर क्या होता है
इंसिडेंट हैंडलिंग. इंसिडेंट
ओके
आपको कापा उसका लेना है जो डायरेक्टली और इनडायरेक्टली प्रोडक्ट को अफेक्ट कर रहा है. सीरियल नंबर गलत हो गया, एंट्री गलत हो गई है, या फिर आपने डाटा इंटीग्रिटी कर दी है, फॉर दैट यू हैव टू लॉग द इंसिडेंट एंड अ ट्रेनिंग डॉक्यूमेंट. दिस टू आर important things. Then various QMS softwares are being available in the market. First one, document control. So document control में होता क्या है? Document control means whenever you draft any document, you you are having the ownership that document. Suppose every organization is having quality assurance, quality control, R&D, production, warehouse, and so many departments are there. So if the particular document belongs to the testing part, then the quality control is the ownership of that testing specification, procedures, and the test parameters. So whenever other department or the cross-functional team members like production or quality assurance needs that document, then it should be issued as a controlled copy. Every organization should be having a system how to draft, how to control, how to issue the particular document. Sometimes we have to distribute the documents apart from our organization to other, likewise the regulators, likewise the auditors. At that situation, we have to issue. We are not going to issue the master documents. Master documents, it should be under the ownership of the particular department, and other department must be issued as either the control copy, either the uncontrolled copy, either the reference copy, or the just for the review purpose. So a particular stamp must be there. So this is under the document control system. Then change control system. Okay, you are having a systematic approach in your organization. So introduction of any new things within a set organization through change, through any particular form, is called as the change control. In change control, we have to analyze the risk of this installation, qualification, and performance of that particular product or the machineries. It may be a kind of document, it may be a kind of document, it may be a kind of machine, man, or material. So different kinds of change control system are there. Then audit management. There are different audits like internal audit and external audits. Every organization should have a system for the internal audit management. So by the following this audit management system, we can say that particular department is following particular system, procedures, policies as defined in our predefined specification. And if during the review of this audit, if auditors found some non-conformances that they used to issue the NCs to particular department, that particular department has to resolve those issues, and they have to submit all these NCs as a NC improvement. Without submission
Excuse me,
of
madam.
this,
Excuse me. You are speaking about this quality management system. and your time and again you are saying about according to the industry, according to the requirement, SOPs all will change.
Hmm.
So, does it really carry the significance of maintaining quality management? Is
Yes,
there any
definitely.
specific guidelines are there in order to be followed, especially to the industry?
Okay, each and every guideline is being prepared by the particular organization. First, there are two types of guideline. One is the guideline issued by the
Organization individually they can modulate that,
just
or
a minute.
else,
Just a minute. I'm going to
yeah.
explain. The first guideline being issued by the regulatory agencies. Okay, you have to follow these. Likewise, ICH, International Conference on Harmonization, WHO, TRS. So these are the regulatory agencies. Some are the regulations being drafted by the quality persons. Quality persons means someone is if you hire some consultant, they come and visit and they found some non-conformances. So they defined the non-conformances, that these are the non-conformances which directly or indirectly affects the your system, directly or indirectly affects the product quality. Again, the third one, suppose a top management review meeting. There must be a system that management should review all this system at least twice in a year. Some in my organization we have a MRM once in a quarter. So in every quarter we are revising all these systems. We put all the suggestions, all the non-conformances in this MRM. We define that if we follow these guidelines, then definitely it will impact these documents. It will impact this departments, and we outlines all these things, submit those, and implement it as per the requirement. So there is no such guideline that you have to revise the document within single time frame. You can revise each and every guideline depending upon the requirement. Requirement of what? Requirement of the regulatory agency, requirement of the quality agencies, requirement of your organization. So once you draft the document, obviously you have to frame a particular time frame when you are going to revise the document, but it should be revised each and every time on the requirement basis. Then the risk management. Quality management system can't be driven off without the risk management. So everyone should be define the risk management of the uh, system. Then training management and vendor management. Again, the quality risk management. Quality risk management, it means to train the person to walk on a tightrope among the dangers. As the picture shows, that a person should be trained to walk on a tightrope among the dangers. How it is should be, how to make the balance. Then these are the perspective. It is defined as a combination of probability of occurrence and harm, that severity of that harm. Then these are the silent features, that always keep end goal in mind, that is the safety and efficacy of the product. It should be as per the customer requirement, as per the regulatory requirement. Then there are some methodologies how the risk has to be estimated, like the comparison matrix, risk estimation matrix, failure mode analysis, faulty tree analysis, HAZOP, PHA, and so on. So I have summarized some of the risk assessment methodologies. By using these methodologies, you can identify the risk.
Can you explain the FTA?
Sorry, faulty tree analysis. Okay, you for this you have to take the fishbone analysis, and in this man, material, management, machine, and parameters. So you have to identify for what reason this non-conformance is observed, whether it belongs to material management, whether it belongs to machine management, whether it belongs to man or something environment. So by drafting this tree, you can say that these are the reasons we have identified this, and this to be overcome to get the analysis.
In
4M,
FTA 4M ki 6M?
4M and 6M both are. You have to question the person till you get the answer. Then risk evaluation and mitigation strategies, FDS perspective. So It is required in risk management plans, the rules, risk minimization strategies, and medication to the patients. So these are the classification, how the risk is being classified as the negligible, moderate, critical and catastrophic, and another one, the probability, almost certain, likely, possible and rare, unlikely. So I have given the number between 1 to 10. You can give any numbers, it depends on you. You may give the numbers from 10 to 100 or 20 to 50, it depends, totally is your choice. But you have to identify the greater number as on catastrophic, less than that in moderate and negligible. Likewise, the three colors in signal, red, green and yellow. These are the parameters levels. If the 15 to 20, then it is extremely critical. It is 8 to 14, then it is high. This is the risk is unacceptable. 4 to 7, it's moderate or medium. And if it comes under 1 to 3, one it's low. Low means it doesn't mean okay, you can regular basis. Yes, the risk is acceptable, but for the time being, it will not be accepted for the long term. You have to identify the risk, plans for further reduction of the risk, and should be implemented with the other security upgrades. Then we draft a table consequences up, against the likelihood. Again, negligible, minor, moderate, major, and catastrophic. If it comes under almost certain, the ranking is 1 to 5. Then you have to multiply likelihood to consequences. 5 is to 1, moderate. If you multiply 5 is to 2, then 2, high. 5 is to 3, 15. 5 is to 4, 20. And 5 is to 5, 25. So based on the numbers you receive, by multiplying this, you can define the risk classified as a negligible, minor, moderate, major and catastrophic. Here I have given number 1 to 5 for the negligible, minor, moderate, major. Again, almost certain, likely, possible, unlikely and rare. These are the justification. Negligible. When we say the risk is negligible, when it will not result in harm acquiring the retention, when it is called as a marginal, results in customer inconvenience and the harm requiring local first and treatment. When it is to be said as a critical or catastrophic, it may result in extensive harm or sometimes it may lead to death. So it should be identified and negligible. It should be identified and overcome for the next products. Again, the rare, likely, possible, unlikely, it defines as a it may occur every 10 to 30 years, 5 to 10, 1 to 5, once in a year. So based on the occurrence, you can define that this risk is unlikely or likely, marginal, moderate or critical. QRM process, it's a four-step process. Very first step is you have to assess the risk. Then after assessment, you have to plan something that can control the risk. Again, it should be communicated and reviewed. In broad way, we can say that QRM process is ACCR. A means assessment, control, communication and review. Here I have tried to explain the risk by putting this figure. So what do you think? What is the risk? High risk, moderate, low or negligible? We have four: catastrophic, critical, then marginal or low risk. What is the risk? Here is the catastrophic. Okay, it is extreme high risk. So in this picture, it shows that the person enters the lion's cage without properly analyzing the risk involved. By doing so, the person's life is totally depend upon the lion's mood. The lion may choose to eat the food by the person, or it may decide to make a meal out the person instead. This careless action puts the life of person in a serious danger. So here in this case, this risk is catastrophic. Hmm? Just इसके पहले वाली स्लाइड में I have defined this as, yeah, this is the checkerboard. I have given the number between 1 to 5. Okay. So on the basis of that estimation, I am going to analyze the risk. So here I have defined seriousness versus possibility. Seriousness 100%, possibility 100%. It totally depends upon lion's mood, what to eat. Then again, in the second figure, the person identifies the risk. He wears the PPE kit. PPE kit means personal protective equipment. And he enters the cage, tries to feed the lion. Here, the seriousness is little bit lower than the previous one. So we may say that seriousness is 4 and but possibility is 100%. So seriousness into possibility is 20. Here, the risk is analyzed, the risk is identified, but proper action still not taken to avoid the risk. So it under the category moderate, right? Again, so we can say that if we are properly analyzing the risk, but we are not identifying how to mitigate the risk, then again it comes under the moderate risk. So in third picture, what you say? What is the risk? Catastrophic, critical, or lower? Why lower? He is outside the cage. So in this picture, the person identifies and analyzes the risk associated with this animal. So we can say that the person taken some precautionary measures. He reduces the size of the gate. See, in the first picture, the size of the gate is bigger than the latter one. Here, he has reduced the size of the cage and he tries to put the food for the lion with this small window. So we can say that he has reduced the size of entry gate, is providing the food to lion through a small window, and this limits the direct contact of the animal with the human being and the chances of accident. The risk still exists but lesser than the previous two. So here in this case, the risk is lesser than previous two, right? Then in the last picture, what do you say here? Here, zero risk. Why? He is outside the cage. He put the food bought for the lion and immediately locked the doors. So in this case, we can say that he assessed the risk, controlled the risk, then communicated in, and finally it is being identified. So we can say that in this case, the seriousness is 0, possibility is 0, and here the risk is 100% reduced. This is how the manufacturing company of the medicine works on the risk assessment matrix. So we can say that it is completely mitigated. It is mitigated through a complete plan, and the person has realized the safest method is to keep the food in the cage, and this allows the lion to eat without any direct human involvement. And by avoiding the close contact, the chances of harm is eliminated. This is what we do in our manufacturing industries, that whenever we identify the risk, we have to plan a risk mitigation plan. We have to identify the risk based on these risk estimation matrices, and then finally we reduce the risk. We have a particular documentary evidence that during the manufacturing processes we have identified these are the risks, we have mitigated the risk, and these are our future plan, so as to avoid such contamination in the future products. So clear everybody how the risks are being categorized: catastrophic, critical, major, and minor. Then five steps of the risk assessment. The very first thing, identify the hazards. Identify the people who might be harmed and how. Evaluate the risk and decide on the precautions. Second one, record the significant findings and implement them. And finally, review and update as necessary. So, as informed earlier also, we have to make a documentary evidences for each and every activity for the retrospective recovery of the data. So these are the silent steps of the recovery. Severity. Severity is directly associated with the patient safety and efficacy. Then occurrence. It is directly associated with the products and the processes. Then detectability. It is directly associated with the measuring or the analytical methods. So we can say that in a nutshell, risk assessment requires brainstorming and meaningful execution. For these are the potential applications. As a part of production, as a part of quality control, as a part of packaging and labeling. The recent NCs received for the non-compliances of quality management system to the pharmaceutical industries. So I have not mentioned the name of the organization. I simply put the date. In October 9, 2025, FDA inspects the organization number A, which found the NC like the failure to establish the procedures to prevent the microcontamination in sterile products. It comes under the quality system control and issue. On October 10th of 2025, again, it inspects the organization B and found that the firm's quality system doesn't conform to CGMP. In October 2027, FDA again issues a warning letter citing the inadequacy in quality system and poor data integrity, a lack of improper CAPA and corrective system. In the month of November, they issues NCs for the testing of incoming component. The quality unit failed to ensure the component identity. In November 4, abroad CGMP non-conformances were control over the manufacturing. And on November 13, API manufacturing issues, they failed to submit the data of the OOS. So here, I'm going to conclude the session of the quality management system and the quality risk assessment. So, if we summarize the today's topic, a proper QMS helps an organization to consistently improve the product quality and gain customer trust. When the QMS practices are followed correctly, they strengthen the process and ensures that the product meets all the regulatory and safety standards. However, if the QMS is not implemented, then definitely an organization has to face the answers being given by the regulatory agencies, and they have to address promptly in order to survive in the industry. So any questions?
Yes,
I-
ma'am. Um, as you said, you have an experience of 20 to 25 years. Theoretically, this is nice, but I would like to know some of the practical methods or difficulties where you have focused or experienced. Just a simple example will help us to enlighten what's the kind of problem you identified and how you have went through that and how you got the solution for it, like that.
Uh,
Is
see,
it possible?
we have a lot to share. We have a lot to describe here, but the
One
time
example.
is not allowing us. Um, okay, I will try to conclude one example. Recently, we got an NC. I'm going to give an example of my organization. Recently, we have an audit from Anvisa Brazil. So, a very small NC, we can say very negligible NC, that the sweeper or the mopping person, he don't know about the quality policy. What is the quality policy of the organization? Even he was not aware of the cleaning procedures, how it is being cleaned and why it is being cleaned. He was simply cleaning the floor of our graduation area, just like mopping in our regular hall or room in our house. So the regulatory agencies, the person calls that person, that housekeeping person, and ask him to take the mop and show him how he is going to mop the area and what is the procedure. So, and he asked him alone. No one is going to answer. as a backend. So he as a user practice, he took the mop, he took the solution, mopping solution, and he started the mopping. So he asked that person, is it your regular practice to mop this area? He simply said, yes, I mop the total areas with this. So the second question he asked, are you going to change this mopping solution in every area or in every first mop or second mop? He said, sometimes I change, sometimes I don't change. The second thing. The third thing, he asked him, do you get any training how to mop this area? He said, no, sir, I know how to mop the area. So I simply mop the area and it's okay. So the fourth question he asked, how you ensure the area is clean? So he said, sir, the traces are removed. So I thought it is clear. So he said, okay. He then he called a quality person. He called a quality assurance person and he called me also. So, madam, please ensure how you are going to ensure that this area is free from bacterial contamination, free from any dust particles, free from any non-viable particles. How you are going to prove? So we are speechless because the the mopping person, explains all he has learned over the years. So this is the major NC we got in recent Anvisa branch. So just you people tell me what would be the steps. This is a very simple example.
Proper training might have been
Proper
given
training.
before employing the
Second,
employee.
work instructions. Work instructions should be there in a bilingual language.
Monitoring.
So, monitoring. And fourth one, microbial bioburden. It should be analyzed properly and we have to validate the cleaning system of our organization. On the basis of that, we can ensure that this area is free from microbial contamination. This area is completely clean and suitable for the processing.
Thank you.
Okay. Thank you so much.
Thank you, ma'am. Ma'am is also with us in the lunch. So if you have any queries, so you can interact with the ma'am in the lunch. So it will be good. So thank you, ma'am.
So, last words from me to you all people. I just wanted to thank you all listening to the session so patiently, and your time, attention, engagement are truly appreciated. And I have this discussion help deepen your understanding of the topic. Your presence and interaction made this session very meaningful. And thank you once again to Dr. Raman sir for giving me an opportunity to interact with you all people. Thank you so much, sir. Thank you all.
Thank you, ma'am. Now I would request our director, sir, to kindly give the certificate to our speaker. Thank you, sir. Sir, you please in the dais. So now I would request our next speaker, Dr. N. Murugesan, sir. Did he in pharmaceutical chemistry, doctorate in the medicinal chemistry. He was the part of the Central Indian Pharmacopoeia Laboratory, Ghaziabad, and also the Central Drug Standard Control Organization, Ministry of Ayush, Ministry of Health and Family Welfare, Government of India. He was the director of Drug Testing Laboratory, New Delhi, Central Drug Testing Laboratory, Chennai and Hyderabad. He has published good number of papers. So I would request director sir to kindly welcome Murugesan sir.
Oh, thank you. Thanks. Good. Okay, thank you.
Sir will deliver his lecture on the analysis of herbal drug by using chromatographic techniques. So I would request sir, please.
For seeing the slides and all those things. Good afternoon, kids. How are you? Huh? Hope you had a very good session. Dr. Purnima has given a very good insight on this ISO and all those things. Okay. I have been asked to give a lecture on this quality control of drugs. Why quality control of drugs? Should we control the quality? Say yes or no. Let it be interactive session. I don't believe in the monologues. I believe in the interactive session. Okay. Good. Should we control the quality or not? Yes. Very good. Why? There should be some reason for it, no? Because you are doctors. We are fortunate to be in the field of saving the life of people. Okay. And moreover, I'm very proud to be an Indian because India is known as the pharmacy for the entire world. We are exporting drugs to 222 countries. Are you not proud?
Yes,
How
sir.
many of you are proud of to be an Indian? Good, very good. Okay, good. In the same topic, I was asked to give a lecture in Bangkok in a WHO meeting. After my lecture, there was a standing ovation. It was not for Dr. Murugesan. It was for our mother India. Okay, so it is our primary duty to be proud to maintain the quality of drugs. Okay, quality is the overall scenario. I strongly believe in RFT. RFT means right in first time. That means first doing something, it is wrong, then we are doing it again. It is a waste of time and all those, So first plan. Planning makes the thing perfect, isn't it? So do it, whatever you do it best. You do the things in such a way that nobody else can do it. Just I was going through this laboratory. In the previous years also, I have visited this laboratory. It was not like that. Now, Dr. Raman Singh has given a golden touch. The Maida's golden touch, just a story you might have heard about, no? Just like that. Similarly, I expect each and every one of you to do that kind of magic touch in your institute. Will you? That's good, very good. That kind of thing should be there. And, I was working as a junior scientific assistant in Indian Pharmacopoeia Commission. Previously, I was known as the Central Indian Pharmacopoeia Laboratory. By that time itself, I was having a vision that one day I should become a director. I was the director of 3 laboratories: Delhi State Testing Laboratory, Chennai Laboratory, and Hyderabad Laboratory. Similarly, if I could do something, you can do better than me. Okay, good. Now let us come to our topic. Quality control of drugs. The quality control, as I told you, in the Drugs and Cosmetic Act, Schedule L is there. Schedule L talks about GLP. Easy to remember. L, no? Schedule L is for GLP. M, Schedule M for manufacturing. Okay? GMP. So GLP is a part of GMP. We are going to talk about the GLP. Some of you are from the manufacturing side also. So GMP is there. You should know something about GMP and GLP also. And then, as I told in the opening session, we have cure in our Ayush drugs system better than in the allopathic system. Okay, so to standardization of this Ayush drugs is more important than in the allopathic drugs. Okay, because the allopathic drugs, either it is a combination, a single drug, or FDC, fixed dose combination. 2 or 3 combination will be there. Whereas in the case of ISM drugs, there will be list, very long list. Have you ever seen any formulation which contains less than 10 drugs? No, isn't it? So in the case of Chyawanprash, name itself indicates 54 ingredients are there. Can any one of you identify each and every ingredient? It is not there. Nobody can do it. Nobody does it also. We have to do it. For that, we have facilities here. As I told you, I was going through the rounds. They are having the best instruments possible. Make use of these 5 days. Okay? First in the morning session, the theoretical session will be there. In the afternoon, hands-on training will be there. So make use of it. Some drug inspectors are also there. Now, so you see that thing. Whenever you go for inspection, you see whether they are following all those standards or not. Okay, good. Now, for quality control, I told you first do it, follow the GMP. If you follow the GMP, at least 90% of the faults can be rectified. Okay, then Madam Purnima was telling about talking about the GLP, QMS, and all those things. No? For that, very simple thing is that do what is written, write what you do. If it is not written, you have not done. Okay, so follow those procedures. Then, if you follow that 90, I told you 90% can be done. For remaining the 10%, that talks about the quality control. Then quality control can be done using three methods. One is spectrophotometry, then chromatography, and another one is miscellaneous instruments. Now we have a mixed audience. Doctors are there, drug inspectors are there, some quality control people are also there. So to maintain balance amongst all of you, I talk in some layman's language. Of course, you may be knowing it, so everybody can understand. You have studied it in your UG or PG, but I would like to refresh those knowledges. Okay, spectrophotometry. The name itself implies. Of course, I don't need any slide. Okay, good. Then, of course, I prepared the Indian Pharmacopoeia IP 1996 as my own PhD thesis. I prepared right from drafting, writing what's called drawing the structure of the chemical formula, printing, binding, everything was done by me in IP 1996. So I talk more about this IP. IP is my child. Okay, in the IP, I see IP before 1985. We had a mixture of herbal drugs and chemical drugs were there. In IP 85, only chemical drugs were there. Of late, now we included herbal drugs in Indian Pharmacopoeia also. Now there are 170 drugs. In that, mostly the HPLC, TLC, and HPTLC are being used. I was talking about the herbal drugs. You have been using it. So, best exam. And one more thing, in the morning, Dr. Raman Singh was telling, one herb, one standard. Why he was saying like that? No, the Tinaveli Senna sites and all those things are there. No? Well, first, it was introduced in Indian Pharmacopoeia. Then the British Pharmacopoeia also introduced that standard. But standards are different. And then the exporters got confused whether we should follow IP or BP and all those things. Now they're harmonizing it. Okay, whether it is allopathic drug or Ayurvedic drug, instrumentation is same. Before that,
sir.
sample preparation,
Excuse me,
that
sir.
matters. We have
Sir.
to do the extraction and all those things. After the extraction, see here, somewhere dry extraction, somewhere extraction with polar solvents, non-polar solvent. I will tell you what is polar solvent and non-polar solvent. See, in the case of water is the best example? Yeah, H plus H2O, no? What is H2O? H plus and OH minus. Two poles are there. So it's a dipole, they are polar solvents. And then if you see cyclohexane, there is no plus. Plus or minus is not there. It's a non-polar solvent. So we extract the drug with either polar solvents or non-polar solvents depending upon the nature. Whereas with the amino acids, we can extract with the water itself. It's the aqueous extract. Because amino acids are freely soluble in water. Whereas if you go for alkalides are not freely soluble. Not at all soluble. Why they are called alkalides? Ravolfo alkalides are there, na? Ravolfia serpentina, we extract it. Why they are called alkalides? They are alkali-like. That's why they are called alkaloids. They are not soluble in water. Then we extract with acidified methanol, that is 0.1 methanolic HCl, or some cyclohexane and all those things we do it. So first they are getting extracted, and then we separate it using chromatography technique. First, let me complete this spectrophotometry, and then I'll go for chromatography. I'll come back again here. Okay, in the herbal analysis, excuse me, you may be very important, but my time is more precious than yours. Please switch off your mobile. Okay, thank you. Okay, in the quality control, while doing standardization drugs, we do the microscopic or macroscopic or physicochemical tests, and then instrumental method of analysis. As I told you, sample preparation is more important. In the ISM drugs, sample not only important, it is very tedious also. If you are already doing it, and you can very well understand the importance of it. Okay. Then sample preparation, how to do it? First, you see whether it is soluble in water or not. If it is insoluble, then go for polar solvents, and then non-polar solvents. Like that, a series of tests need to be done, and then you do it. Okay. Now, as I told you, there are three major classifications for drug analysis. Three, as far as instrumentations are concerned. One is spectrophotometry, another one is chromatography, another one is miscellaneous. Spectrophotometry. Photo means light. Everybody knows. Is it not simple? Photo means light. Metry means measurement. So photometry means measurement of light. Spectrum means with a wide range. So spectrophotometry means measurement of light in a wide range. Okay. Then for that, why to measure the light? Because certain drugs absorb some light to go to higher energy level. This absorption of light depends upon the concentration of the solution. There was two scientists. First scientist Lambert, another one is Beer. Lambert said, when a beam of monochromatic light is passed through a homogeneous absorbing medium, the intensity of transmitted light is directly proportional to the thickness of the medium. This is Lambert's law. Then Beer slightly modified it. Instead of taking the glass medium, he took solution. Then he said, when a beam of monochromatic light is passed through a homogeneous absorbing solution, the intensity of transmitted light which comes out. Incident light falls in first, and then it absorbs the light, then the remaining light transmitted. The intensity of transmitted light is directly proportional to the concentration of solution. Thereby, we measure the concentration of solution, concentration of drug in that particular preparation using the spectrophotometry. Then, again, I would like to take you to your school level, in the 9th standard or 8th standard, okay, where you might have studied the WebGR. Sir Isaac Newton, when he was a young child, he was playing in his room, playing in his house. It was having thatched roof. Thatched roof, you understand? No? Thatched roof. Some sunlight was coming through it. And he was having something in his hand. As a child, he was curious enough to play with it. Then something which he was having was nothing but a prism. Okay. When the prism was kept in the sunlight, it disposed into 7 colors. That violet, indigo, blue, green, yellow, orange, red. These colors you can see, no? Can you see these colors or not? Violet to red. These colors you can see. Can you see? Is it visible? Okay. So it is visible colors. So if you measure the color intensity, then it is visible spectrophotometry. Okay, understood? It starts from violet and ends up to red. So this color, the intensity of the color is directly proportional to the concentration of the particular drug in it. So when we talk about visible, there are some invisible also, no? Isn't it? So before violet, it started with violet and ends with red. Before violet, something is there. Similarly, after red is also something is there. Though we can't see this particular color, using instruments we can measure it. So before violet, in Greek, ultra means before. So it is ultraviolet spectrophotometry. Understood? Then after red, again in Greek, after means infrared spectrophotometry. So in the electromagnetic spectrum, right from ultraviolet spectrophotometry, then visible spectrophotometry, then infrared spectrophotometry. Above that also, some metals cannot be detected even by the IR. So it is NIR, near infrared spectrophotometry. So today we are going to talk about all those things. Can you understand all those things? Huh? So you understand this visible. Certain colors we can see. It is green color, it is brown color, that we can see. So whatever color we see, it is visible spectrophotometry. Before visible, that violet, it starts from violet to red, VGR. So before violet, ultraviolet. After red, infrared spectrophotometry. And then, so these are the various spectrophotometers. Whatever I am talking now, they are the instruments. They will give you hands-on training also, how to use those instruments. And then, one more thing I would like to tell you. There is one atomic absorption spectrophotometry. That also I'll explain. I don't know, I will in time or not. FTIR is the then mass spectrophotometry. And then this laboratory is very rich. They are having GCMS for pesticide analysis. They are having GCMS and MS. They will give you the demo of this thing also. What is mass spectrophotometry? I told you about the UV visible IR. Then atoms or molecular spectroscopy is there. Certain atoms absorb light and go to a higher energy level. Then you, or I can give you again layman example. See, now you are there. You are working in government. You will be getting around 3 to 4 lakhs rupees salary per month. Okay. In another 5 years, and now you are getting 3 lakhs. So your life started, you know very well. Okay. In another 5 years, if you get 30 lakhs rupees per month, tell me one thing honestly, will your lifestyle be same? You will buy a new car, new bungalow, and all those things, isn't it? Similarly, the atoms or molecules, when it absorbs some light, it goes to a higher energy level. This rise in the level is directly proportional to the concentration. So there's like salary. When you get 3 lakhs, one level. 30 lakhs, another level. Like that. If concentration is less, some level is there. If concentration is more, then higher energy level. Similar, based on that energy level, we measure the concentration of the solution. Okay? And then we can see whether it is there or not. And the identification. Identification, yes or no. Okay? Positive or negative. That we see then. When it is positive, then it goes to, we go to higher energy level. Okay. The spectrophotometry, just light outline I have given, because spectrophotometry itself is the ocean. Now, if you ask me to talk about spectrophotometry, I'll talk for at least 3 days. Now, apart from MSc, PhD in medicinal chemistry, I have done diploma in instrumentation also. I have a special liking for instruments. I like those instruments like my own child. Now, it's okay. Then chromatography. Again, simple layman example which you can remember all those. Chroma means color. Don't think that I am underestimating you. I would like to keep something. I would like to inculcate something in your mind so that you can never forget. Chromatography, people think it is very tedious and all. It is not so. Chroma means color. Graph means presentation. Okay. Chromatography means based on the color, separate it and represent it on a paper. That's chromatography. Is it not simple? Good. Why chroma? Previously, the herbs or herbal drugs were separated based on the color. Very simple example is chlorophyll. Everybody knows chlorophyll is green in color. Okay, just dissolve the sample in water, filter it. You will have a green color solution. Okay, that means all the chlorophyll has come. Similarly, based on the colors, the constituents, different constituents of a mixture were separated. Okay, then you can ask me one thing. Can you analyze only colored compound is in the chromatography? No, it is not so. Even colorless compounds also can be analyzed. What is the first definition you studied in chemistry? During your school days, you might have studied in chemistry, no? Okay. What is the first definition we studied? Atom. Atom is the smallest indivisible particle of an element. Am I right? I studied around 50 years back. Maybe I'm wrong. Huh? Atom is the smallest indivisible particle of an element. That means which cannot be divided. Indivisible particle of an element. Then what is atom bomb? When you divide atom, it gives energy. That means it can be divided. So it is not indivisible. So the very first definition I studied in my schools is wrong. Isn't it? The chemistry, very first definition I studied in chemistry is wrong. Same is the case here also. Not chromatography is not only the colored compound. Even colorless compounds also can be separated. Okay, how? There should be some reason for it. Okay, I am walking from here to here. Okay, oh, because the floor is stable. Only I am moving. Okay, so I am walking from here to there. Similarly, see, when I speak, my speed is little lesser. Otherwise, I can walk fast. Isn't it? So that is something which affects my speed also. Similarly, for separation, we need two phases. One should be stable, like floor. The other one should be moving. So there are two phases. One is stationary phase, another one is mobile phase. Can you understand? It is very simple, you know. So one stationary phase is there, one mobile phase is there. Say based on that we do. And you can ask me one thing, what is the stationary phase, mobile phase, and all those things. Now I know only one mobile, which is in my pocket. Nah, but it is not that one. See, there are three phases we know in the nature. One is solid phase, another one is liquid phase, what is the third one? Gaseous. Very good. Okay. As I told you, one is stationary phase, another one is mobile phase. Let us take three instances. Three phases here, the solid, liquid, and gas. Here, again, this is a stationary phase and this is a mobile phase. Solid, liquid, and gas. The board is not there, otherwise I will write. See, when the separation happens between solid to solid, okay? Solid to solid separation is possible? You know, girls know better. Why am I saying girls? No. See, in home, while you prepare food, if there are small stones, chota pattar, in the rice, what do you do? Just we sieve it. Okay? The sieve is a solid and the matter to be separated, that is also solid. Okay, solid chromatography. It is known as leaching. Okay, solid chromatography. It is called as based on the weight. Small stones comes first, then big stones come later. Based on the weight of the stone, they get separated. It happens, no? Though you may not, then you might have seen. So this is called based on the size. We separate it or exclude it. So it is size exclusion chromatography. Okay. What is which size? Size of the molecule. Molecular molecular size exclusion chromatography. SEC. This we do it in proteins. Okay, this is very rare. Okay, then next one, solid is over. Then solid and liquid. First solid stationary phase, liquid mobile phase. Solid liquid chromatography. Here, again based on technique, the chromatography can be separated, classified as adsorption chromatography and absorption chromatography. You can say absorption, you know very well, because you are absorbed in the particular post. Absorption, you know. What is adsorption? Adsorption, again, example. Actually, I was a lecturer in a pharmacy college, so I give more examples. Now, the example is not to mislead you or take time. You can understand it. Take a tennis ball, dip it in water. Only the external surface gets wet, but inside it is intact. Okay? Similarly, in the case of absorption, when it comes in contact with the liquid, only the external surface gets wet. Okay? So it is adsorption chromatography. You take a cotton ball, dip it in water. It will get wet both external side and internal side also. It is absorption. Can you understand the difference between adsorption and absorption? Adsorption means only on the external surface. Absorption means both sides, internal and external. Is it simple or not? Can you understand all of you? Okay, good. Then the chromatography, we talk about only this adsorption chromatography. Adsorption technique only, not absorption. Absorption, we talk in spectrophotometry. Whereas in the chromatography, talk more on adsorption. Okay. Then solid we have seen. Solid and liquid we have seen is adsorption chromatography. Another one is partition chromatography. Partition. See, now you are there. If you are a single boy in your home, you will get all the shares of your father. If there are 4 or 5 kids in your family, it will be divided among all the division we call partition. Property partition we say, no? So, if partition, if it is among, if the partition is done among the same family, okay, it is partition chromatography. Partition, if there are 5 kids, everybody will not get 20% share. The eldest boy in the family might have worked hard, very far, so he will get more share. Okay, so depending upon the periodicity, the partition varies. Partition. Similarly, certain drugs dissolve more in polar solvent. Polar solvent, non-polar solvent, I told you. Different, or forget polar, non-polar. Dissolve more in one particular solvent. The example is sugar. And all of you know very well, sugar dissolves very well in water, but it's insoluble in kerosene. If you are having a mixture of what's called the sugar in kerosene, then how to separate it? Pour water, shake it well. Now all the sugar will come into water. Evaporate the water, you will get sugar. Got it separated? This is what chromatography. No, chromatography is very simple, no? So partition, based on its solubility or partition. huh, we separate not only one mission, various constituents. For example, in drugs, I told you there are a list of 10 or 15 ingredients in the formulation. All the 15 ingredients can be separated using this chromatography technique, partition chromatography. So now we studied solid-solid chromatography, that is size exclusion chromatography. Another one is solid liquid chromatography, that's absorption chromatography. Another one is liquid chromatography, that is partition chromatography. Is it clear?
Yes, sir.
Shall we go to the third one? Okay, then solid and gas. Solid gas chromatography. Gas chromatography is such a technique where you can separate the gaseous sample and volatile sample also. If you are having a mixture of say methanol and ethanol, and you know very well methanol's boiling point is 68, ethanol 77. So volatilize it, and then based on its boiling point, they can be separated. They will give two different peaks. Similarly, either volatile samples or you can convert them into volatile. For example, my friend is what's the good name? Okay, good. He is there. So now he has come to office. He is in formal dress. No. So whatever I ask him, he will reply only officially. Whereas if I go to his house, he will not be in formal dress. He will be in casual dress. Okay. I don't know him personally, but I know his good friend. Okay. I'll talk to him. Then he will make it amenable. Make him amenable. So whatever I want, I can get it done through him. Similarly, though he is not volatile, I can convert him into volatile friend. So gas chromatography, either volatile compounds or volatile derivatives of compounds can be analyzed using gas chromatography. Understood? You need a much simpler example, or you can understand this thing. Good. You can understand, no? Good. So these are solid chromatography with solid chromatography, solid liquid chromatography, solid gas chromatography also we can see. Then comes liquid. Liquid as a stationary phase. Okay. And mobile phase is also liquid. This is liquid chromatography. This is also partition chromatography. I will give example for each and every thing also. Then gas liquid chromatography. Okay. Liquid is a stationary phase. Gas is a mobile phase. So gas liquid chromatography, GLC, gas liquid chromatography. Generally, we talk about when you talk about GLC, it is GC. We don't say Ghaziabad city and all those things. When you say Ghaziabad, name itself implies it is a city, isn't it? So when you say GC, it is GLC, gas liquid chromatography. Can you understand this basic? Okay. Shall we go to the technique now? Okay, good. So here we have a sample. As I told you, sample preparation. In the case of allopathy drug, no, we just extract with the single liquid and then we do it. Whereas here in the case of Ayurvedic Indian system of medicine, we have to do multiple extraction, sometimes steam distillation also we have to do. And then, right. Then I was talking about the speed also. Why I was talking about speed? No, the reason is affinity. Okay, is alone. Now I ask him to walk from here to here. He will walk in a particular speed. When his good friend goes with him, he will walk in a different speed because they will be chit-chatting. When he is a group leader, so 10 people are going with him. There will be difference in the speed or not in all the three cases. Similarly, one compound is there. Okay, it will be having some affinity to it, to that compound, having some affinity towards the stationary phase. I told you, stationary phase. There are so many stationary phases. One is silica gel. Silica gel is mostly 90% silica gel is being used as an adsorbent. Why it is silica gel? Gel, you know. What is gel? Everybody likes gel, no? What is that gel? Ice cream? You like ice cream? Say yes or no? Huh? Ice cream, you scream, everybody scream for ice cream. Na? Uh, so, ice cream is the best example for gel. One liquid particle surrounded by so many solid particles is a gel. Okay? One solid particle surrounded by so many liquid particles is sol. One liquid particle surrounded by so many solid particles is gel. This sol gel transformation, we call it thixotropy. When you go to beach, when you put your feet, some water comes in. When you take your feet, then water goes away, fades away. Okay, thixotropy means sol gel transformation. Yeah? So water comes out of the sand, and then we take your feet, then water fades away. So this is so gel is the best example. In compared to the powder, in gel form, separation happens better. In the molten stage, you can say. So in case of gel, separation happens better. That is our my ultimate purpose, no? To separate the different constituents of a mixture. Okay. So we take silica gel. So first we dissolve the sample in some solvents and then put it in the chromatograph. Then it gets separated, it comes out. Now all of you are here as a trainee. You can be classified on various bases. For example, I can say gents, ladies, or people from state government or central government, or those who are already in the quality control field or from other fields, or doctors, non-medical doctors. Okay, where is separation classification? Same people I can classify on different. Of course, I don't believe in dividing policy. Dividend policy, I don't believe in at all. Whereas people can be classified like that. Similarly, classification of this different constituents of mixture can be done on different things. First, sample preparation is done, then separation done on the chromatography, and then when it comes out, I have to identify. She is Dr. So and so. He is Dr. So and so. So identification. Okay. So in the chromatographic instruments, what are the three essential? And then I have to record it also. I have a very poor memory. So I am seeing here today and seeing you today. Tomorrow, if you ask me, then I'll forget. No. So forgetting is a human tendency, isn't it? So we keep it in record. Okay. As I told you, do what is written, write what you do. So you have to keep the record of everything. That we do know. The same thing happens in chromatography. In a nutshell, I'm telling you again. First, sample preparation. When the sample should be injected in the chromatogram, so we need a sample injector. Then, for separating the things, we need a column. Then, after it comes out, we have to identify the compound. So, detecting. So, we need a detector. Then, after detecting, we have to print it somewhere. Printer, plotter, or recorder. We need it. These are the four constituents of a chromatogram. Okay. So, study is chromatography. Technique is chromatographic technique. And after separation, the data you get is chromatogram. Understood? So, these are things of a essential chromatogram instrument. Okay, good. For your convenience, they already told you the mixture of mixture you're having. Now, then they get separated based on the affinity or partition you get. The component get see in the initial first one, the green, pink, blue, the violet, everything is there. Then they get separated. So this is the very purpose of this chromatography technique. Okay. um, Stevenson coined the term chromatography in 1917. So it is more than 100 years old technique. Okay. He said um, components can be separated based on the color. And I told you, even colorless compounds also can be separated. Okay. Now, shall you put it in simple words? Chromatography definition. Can you ask me one thing? First definition should be there and then explanation. But if I tell you the definition in the initial stage, you may not remember it. Now you can remember. I'm not saying you can't understand. You can very well understand, but you can remember. So the very purpose of this chromatography is to isolate. Isolate means separate. Okay. Second one is identify. You identify, no? What is that? This is what is alkali, typewriter, and all those you can. Then quantify. The alkali, Vidania somnifera, isn't it? That alkali separation if you do, then there are 21 alkalis in the Vidania somnifera. All the 21 you can get separated. So first isolate. Isolate means separation. Then identify. Then quantify. So now can we redefine the chromatography? Chromatography is a modern technique to isolate, identify, and quantify different constituents of a mixture. Can you remember the definition? Can you remember the definition of chromatography? No. No. Your face is. I have done MBA, so I can read your face like a book. So chromatography, very purpose of this instrument is to isolate, identify, and quantify the constituents of a mixture. So this is the instrument. Okay. Then morning, Dr. Raman Singh was telling that about the TLC, HPLC, HPTLC, and all those things. Again in chromatography, now you will understand a little bit of chromatography, no? Okay. Then, because I have slides for everything, but you are my good friend, so I can interact with you face to face. Then, again classification, one more classification is that. Again classification, those who are wearing blazer or not wearing blazer. This is just a simple example. Don't take it otherwise. Okay. Similarly, chromatography, the separation, identification, isolation, identification, quantification can be done in a different way. The different way means the classification can be done in a different way. How? The process, right from isolation into quantification, whether it is happening on a column or on a flat bed. Okay? So if it is done on a flat bed, it's called a flat bed chromatography. If it is done in a column, it's a column chromatography. Is it not simple? Name itself implies. Okay, flatbed chromatography. What is a flatbed chromatography? Example, paper chromatography or thin layer chromatography. Because I don't read it. Users. This will come to you later. These are various chromatography. Okay. I was talking about flat bed chromatography. Okay. Flat bed chromatography. Paper is flat. Okay. The separation can happen on paper. So it is a flat bed chromatography. Okay. Then, so classification is either flat bed chromatography or column chromatography. This separation you understand? Okay. First, let us see the paper chromatography. Paper chromatography, again, based on ascending technique. Whether you go by two-wheeler or four-wheeler or helicopter or flight, just like that. Based on the separation technique, again it can be classified. And it is a paper. You keep a solvent here, keep a sample here, then allow it to rise. Okay, then it goes up now. So it is ascending chromatography. Simple, no? It is ascending chromatography. In certain cases, if the extract, plant extract is so thick, it will not go up. It then keep the solvent up. Apply your spots here. Then descending chromatography, and then it comes down. It is descending chromatography. Best example is analysis of amino acids. What are amino acids? Doctor friends can tell me better. Amino acids. Amino acids are the building blocks of our body. Amino acids combine together to form proteins. We are made up of proteins. That means we are made of amino acids. For separation of amino acids, the best solvent system we use, bar system. Butanol, acetic acid, water. 4:1:1 ratio. That we use it. Okay. It is a very good technique, very versatile technique. Unfortunately, the technique is being used only in colleges or schools to explain what is chromatography. Okay. So now we studied ascending chromatography, descending chromatography. Understood? समझ में आ रहा? हाँ? Actually, my Hindi is very poor. Of course, I learned Hindi from Ghaziabad only. So, being a Tamilian, we were not permitted to learn Hindi. The CIP, Central Uniform Laboratory, it is in Sector 23. I was not knowing what is Sector 23. 23 I was not knowing. Then, actually, in Tamil, just like 23, no? In Tamil also, you say like that. 23, like that. I thought the same yardstick I can apply here also. So like he said, B is thin. Then they could understand. He took me, I came with auto, took me to sector 20, then sector 3, and all those I could understand. Huh? So at last I learned Hindi. I was forced to learn Hindi. Now, of course, I'm a little fluent in Hindi. I got so many awards also from the Hindi Parliamentary Committee also. So during exposure, we will learn everything like that. Here, paper chromatography is a versatile technique, where it is used only in the colleges or the school to learn what is chromatography. Okay, the ascending technique I told you, descending technique also I told you. And then one more thing is circular chromatography is there. Okay, circular chromatography means it is a precursor to gel electrophoresis. Okay, this you can do at your home also. Take a round filter paper, prepare a wick, put a drop of ink over it, put it in water in a beaker, then water will rise due to capillary action, and then it will spread. The ink will give 7 colors, methyl violet, like that, crystal violet, and all. It gives 7 different colors. That separation happens. No? So it is a paper chromatography. In paper chromatography, what do I do? Ascending chromatography, descending chromatography, and circular chromatography. Understood? So next, let us go to the other technique. TLC. TLC. What is TLC? Thin layer chromatography. It can be called thick layer chromatography also. That is also TLC, no? Why thin layer? Aji, why can't it be thick layer? Actually, we always believe in bulk, no? Salary also we want to get in bulk. Nah, Aji. Okay, sure. Okay, जी अच्छा जरूर. I want to get. हाँ, ठीक है. This thin layer chromatography. Now, HPLC is also there. In the first slide you had seen, the sample preparation was put through a syringe and then gets separated. Okay. During my PhD period, we were using that technique only. This, in a column, we were taking adsorbent. I told you silica gel. Then our sample was kept at the top using a cotton, and then different solvents are poured. Then components get separated. But it was taking a lot of time, 3 or 4 days. Nowadays, nobody is having that much time and patience. So we put some pressure on it. It was getting eluted soon. Okay, so that is called the illusion. Better illusions come, no? So better performance. That better performance is due to the high pressure we give. So the high performance liquid chromatography, I can say, or the high performance is due to high pressure, I told you. So I can call it as a high pressure liquid chromatography. That instrument is very costly. 40 to 50 lakhs rupees in any case. So can I call it as high priced liquid chromatography also? Okay. So this is HPLC. In HPLC, we can do both adsorption chromatography and partition chromatography also. Adsorption means solid liquid chromatography is adsorption chromatography. Liquid chromatography is partition chromatography. So if a sample gets separated in this liquid, this partition chromatography. Then these are the technique. Then I told you it's very costly, 40 to 50 lakhs. Sometimes it goes up to 1 crore also. That is due to the detector. I told you sample preparation. Second one is column for separation. Third one is detector. First two I explained. Third one I am coming. The elution thing. After elution, the eluents. Eluents means whatever comes out of it is eluent. So the time taken for your sample from injection to the detector is called as retention time. How long it is being retained in the column. It is retention time. I told you the walking speed and all this. So based on the walking speed, people can be separated. Similarly, based on the affinity of the sample towards the stationary phase can be separated. It's the retention time. Based on the RT, the samples can be separated, quantified, and then it can be recorded. Detector, I told you. Various technique I told you. The spectrophotometry I told you. No? Here, the spectrophotometers are being used as a detector. There are fixed wavelength detector and multi-wavelength detector. When we started HPLC long back, that is, we were using only the 40 years back, only 2 wavelength detector. One is 254 and 3666. But later on, it converts into different things. Dr. Adawar is here. He is my Guruji. Actually, I worked under him. So he gave me full freedom to learn the instrument. Whatever I am today, it is because of him only. Thank you, sir. I take this opportunity to thank you again. And then, sample preparation, you understand? Separation using column chromatography, you understand? Then detector, UV detector, single detector or a multi-wavelength detector. Then sometimes we use MS also, mass spectrophotometry. But it is unknown compound. I know who are you, so I can call you by name. But if I don't know, then I can say the trainee from wearing cap. So again, what kind of cap you are using? Based on I can separate like that using mass spectrophotometry. When it is a known compound, using a UV detector and all those you can do. If it is in traces, now you are only one among 50. Again, I very well identify you. If you are one among 5,000, very difficult to identify. Okay, so if it is very traces, very less quantity, or if an unknown quantity, we go for MS. So it's a liquid chromatography with mass. Now we believe in hyphenated technology, LCMS, LCMS, GCMS. And I told you they are very rich. They are having GCMS, MS. Okay, that also can be done. Now they will give you a demonstration. So I was giving them basic structure. Okay. So here we inject the sample. Different solvents get pumped to give high pressure, and then goes to the column. Then the element comes out. The elements are detected. So these are the basic schematic diagram of HPLC. The various HPLCs are there. They are having around 30 to 40 HPLCs, I think. So retention time, you understand no? How long the sample is being retained in the column. This is retention time. Based on time, the samples can be separated. The application where it is being used. Now we are in the medical field, so pharmaceuticals. In environment also it is being used. This I already told you. UV detector, very detector I told you. Multiple PDA. Photodiode array detector. Photodiode array means, see, UV detector I told you. If it is a single component, can be done with a single component. Whereas photodiode means it is a very several use spectrophotometers. See, now you have come here. At the enter, while you enter the campus, there was a security guard. Chowkidar was there. He was asking, "Who are you? What for you going inside?" and all those things. Only one person was there. Whereas if one minister comes, there will be 100 chowkidars. They will screen you like anything. Okay? What for you coming? From where are you coming? What is your antecedent? Parental thing, everything they will ask you. If the Prime Minister comes to this office, there will be thousands of chowkidars. Isn't it? They will scrutinize everywhere. Similarly, in the grading, it is there will be so many lines. Each and every line acts as a spectrophotometer. Okay? So it will give exact result. For example, if your compound is having absorption maximum of 257 nanometer, only that particular component will be selected. Okay? The other lights are called stray light and all those things. To avoid it, only particular light is given. So that is PDA detector. Understood? The TLC I already told you. Again, so I'll take only 5 minutes. Okay, another 5 minutes or 2 minutes. Oh, okay, good. Okay, good. So I'll cut short my thing. No, TLC. TLC absorbent I told you. Again, thin layer chromatography. TLC I was asking, she was selling thin layer chromatography. It can be thick layer chromatography. Why not thick layer chromatography? Okay, if it is a thin layer, then detection is easier. If you are amongst a crowd, 1,000 people, identifying you is very difficult. If only 3 or 4 are there, I can identify very easily. So that's why we are using a thin layer chromatograph. Okay, yeah, absorbent spread on the plate, and then they get separated. Okay, then again I would like to give one more example. The sugar candy. The sugar candy you take, no? When you take it, the sugar taste will not come to you so soon. Whereas if you take sugar, it comes soon. If you take the powder of the sugar, then we'll get the response immediately. That means the size of the sugar molecule, that matters. So when the particle size is lesser, then it gets separated quickly. So this is HPTLC. TLC already, HPTLC. Okay. आणि HPTLC का demonstration दे दीजिए. Okay. This is already explained all those things. The solvent, keep a sample, then allow it doesn't, then it gets separated. So based on that, you can identify. Identification can be done using iodine chamber or by spraying some liquid. You can get them identified. On HPTLC, you can directly quantify it. That's called in situ. In situ means within the system itself, you can quantify it. That is, it's such a vast subject. See, initially, the first picture, samples are kept, 2 dots are there, no? Standard and sample. Then they get separated. After 5 minutes, they get separated. Then can be the separation is much higher. Resolution, they are resolved. So you can get separate all the 3 constituents of this mixture. Okay. The UV light I told you, retention factor I told you. So the TLC chromatography is being used for all those things. Separation, isolation, identification, and then quantification. That can be done. HPTLC, of course, almost same thing. Only the particle size is different. Okay, so they will explain you all those things. Demo, everything can be done. No? Hands-on training, you know, almost same thing. Okay, good. Thanks for patiently listening.
Sir, excuse me.
He will give you a copy of my presentation to all of you. No. Then you can see the thing. Pen drive.
Sir,
Oh, he's very generous. Yeah, he's giving pen drive. In other training programs, just we collect their email ID and then we send it to by email. So this is the thing.
sir, excuse me, sir.
Just so this is the HPTLC instrument. They are having all those things. They are having the latest instruments also. It's a CAMAG TLC instrument. What's called? They opened a R&D center in Mumbai. Of course, they asked me to inaugurate it. So 15 years back, the CAMAG. The first one will innovate sample application of sample, and then there's a scan after development. They're being scanned. Then for identification, you use this UV chamber, and then the reprostar for documentation. I told you the 4 essential components of the chromatogram. Everything is there. This explains all those things, how to do it and all those things. Because it gives a 3D spectrum. Okay, good. Any queries you can ask me right now, or you can.
Sir.
I have given my contact details, my email ID, phone number, everything is there. He is giving you pen drive, so you will remember my ID. You
Sir.
can talk to me at any time. Okay. I
Sir,
would like to give you
sir,
clarification right now.
Sir,
but
one
he said time is
clarification.
not there.
Sir, one clarification, sir.
Yes.
Yeah, I'm Dr. Sai Sudhakar from Andhra Pradesh, sir. Previously, I worked as professor and principal, and now I'm additional director. I did my PhD in chemistry, sir. Luckily, last year, our vice chancellor, sir, he's neurosurgeon. Now I became a PhD guide to him. His topic is clot. Because he's a neurosurgeon, is there any solution in your science Ayurveda for blood clot in the brain? Then I suggested Deluka Charana is a good practice, sir, because Hiridin is there that may relieve the clot. But which technique is very good to identify the percentage of Hiridin in the blood sample?
Using streptokinase, you dissolve the clot.
Sir.
Okay. Take sample, and then
HPTLC?
HPTLC can be done.
Sir.
And it better get done by the gel electrophoresis.
Sir,
Because,
gel
the clotting is mainly due to protein clotting.
Yes, sir.
Okay, protein
Yes.
clotting can be better analyzed using gel electrophoresis. They are having gel electrophoresis also.
Yeah,
They will
thank
show
you,
you
sir.
that. Gel
Thank you.
electrophoresis.
Yes, sir.
Okay?
Thank you so much.
Excuse me, sir. Sir, initially you said about harmonizing both Indian pharmacopoeia and British pharmacopoeia. I'm having a doubt that the area where the plants have been cultivated in those areas, will the alkaloids and the components which are present will vary or not? If it is varying, harmonizing both monographs, will it affect the quality of the drug, or how it will be
It
affecting?
will not altogether different because the concentration, constraints may be same. For example, philanthus neurulin or Withania somniferum. The alkaloids which present in remain same. So depending upon the season or collection time, if you collect it in the morning, some quantity 40%
Yes,
will be there. If you collect it in the evening, it will be only 4%. So concentration may differ. But the constituents, that particular alkaloid, one will be there, same.
Okay, thank you.
Sir,
Sanjeev.
sir, here. Sir, I am Abhishesh. I'm from Indian Pharmacopoeia, Phyto Division. Actually, I have one question, sir. What happened now that ethylene glycol, diethylene glycol issue happened in Tamil Nadu, sir? It is a method, so many methods we can, is there to detect the, like GCMS like that. But any alternative method, it's there, sir, to detect the, like HPTLC? Like
Good,
that.
very good, very good. See, again, there are two kind of formulation. One is pharmacopoeial, another one is non-pharmacopoeial.
Yes, sir.
If it is official in pharmacopoeia, it's a pharmacopoeial preparation.
Yes, sir.
Okay, in that case, you have to follow the particular pharmacopoeia.
Yes, sir.
If it is non-pharmacopoeial,
Yes,
you can follow any method
yes.
as a government list. You can develop your own method,
One month.
or you can get the method of analysis from manufacturer, because you cannot develop method for each and every formulation.
Okay.
If you start developing method for each and every formulation, you will end up only with the method development.
Yes, sir.
You can never perform your duty. Okay,
Yes,
so
sir.
in that case, you can get the method of analysis from the manufacturer,
Yes.
but you have to evaluate it.
Okay.
Huh?
Yes, sir.
Evaluate it and then validate it also.
Okay.
Validate. There are two different things.
Yes.
First, evaluation whether it works.
Yes, sir.
Validate means it is only for the particular component or not.
Yes, sir.
Okay, so the validate. So 3 or 4 samples, 4 different batches.
Yes,
You
sir.
have to do it.
Yes.
Whether the cells coming are separate, accurate, and all those things you have to see.
Yes,
That's
sir.
validation. That you have to do.
Yes. 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 Ah. Pharmacopoeia. Yes. Yes, sir. Thank you, sir.
Actually, he is a pharmacopoeia man. We call him mobile pharmacopoeia. So
Yes.
all pharmacopoeia is very
Okay, sir. Okay, sir.
okay, good. Thank you. Due to passage of time, I'm cut shorting my lecture. Let us interact more in future. Okay, bye. Thanks, Dr. Singh,
Yes.
for giving me this wonderful opportunity. Okay, good.
Thank you, sir. Thank you very much for your such nice presentation. अब
Thank
मैं
you.
अपने निदेशक महोदय से रिक्वेस्ट करूंगा कि वो डॉक्टर मुरुगेसन सर को सर्टिफिकेट देकर अब मैं डॉक्टर बाइके फ्रॉडर से सर से अनुरोध करूंगा कि वो मंच पर आने का कष्ट करें. Our next lecture is will be delivered by Dr. Baike Frathor sir. He is the ex-director, Central Revenue Control Laboratory, Ministry of Finance. Sir is having very vast experience in the field of laboratory setup. He has served in many organizations in various capacities like chemical examiner, government analyst, joint director, etc. Sir has published many research articles in many national and international journals. And sir is having good knowledge about the enable accreditation process and how you can set up your laboratory and get enable accreditation. So sir, today मैं अपने डायरेक्टर से अनुरोध करूंगा कि सर को बुके देकर सम्मानित करें. इधर से जी जी नहीं इसी से गुड आफ्टरनून टू एवरीबॉडी काम कर रहे हैं दिक्कत तरह से लगा दो
Uh, Good afternoon to everybody. Topic of my is to how to get an international accreditation. Although I think how many of you are from the laboratory? Less number of the persons. Mostly you are from the control side. Okay. Now, drug test laboratory is a facility that conducts analytical tests on drug and cosmetic to ensure quality, safety, and efficacy of the product. And all the drug testing laboratories, they are being approved from the drug control authorities because approval is gives them a legal sanctity, and approval is given based on the mainly I am is focusing on the IUS drugs because earlier there were only Ministry of Health having the both wings Ayurveda and homeopathy and modern medicine, but now the Ministry of IUS is there. So and this the IUS drug laboratory, they get the approval on the basis of their application, which is under section 160 of the drug and cosmetics rules, and they have set up of the actually some requirement, and those requirements have been published by at that time Ministry of Health was there. It was published in March 2013, but Department of House was there. Now the Ministry of IUS is independent Ministry of IUS is there. So this manual has the guidelines for the inspectors how to go through inspection of the laboratory and the criteria which has to be checked by those inspectors for grant of license. So the requirement of the laboratory was this laboratory should have a premise. Minimum required area was 800 square feet. Testing and other sections, technical manpower, equipment, reference document, and reference samples. This was the criteria for the requirement of that document. Now, premise of high drug testing labs should be well lighted, properly ventilated, effectively separated from non-compatible areas, have proper control of environment and temperature, have adequate health and safety arrangements. Whereas sections required for the testing of these IUS drugs, botany or pharmacology, chemistry, microbiology, Ayurveda, Siddha, and Unani, sample section, office come record section, and requirement for the experts. One Ayurveda, Siddha, and Unani, because at that time this homeopathy was not included. Recently, Government of India has included to homeopathy also, so this presentation is not including homeopathy, but homeopathy also has been now under the Ministry of Ayush. So, expert for Ayurveda, Siddha, Unani, and homeopathy. Degree qualification recognized by the under Schedule 2 of Indian Medicine Council, Central Council Act 1970. In the field of chemistry, again, bachelor degree in science or pharmacy, having at minimum requirement as per the for accreditation 5 years. And in the Department of Botany or pharmacy, bachelor degree in science, that is medical physics, chemistry, sorry, zoology, botany, and chemistry. Pharmacy or pharmacy Ayurveda awarded by recognized university. Microbiology, bachelor degree in science, medical, recognized or pharmacy. This was the qualification of the experts actually, who science the test report. And equipment for chemical sections. This is the list of the equipments which are required for the. Although nowadays more equipments are required, but this was the minimum requirement for to run a lab which can test the Ayurveda, Unani, Siddha, and homeopathic drugs. And this is the requirement for the requirement of instrument for pharmacy section. And this is requirement for you will have the presentation of this, so you can have the go through the because shortage of time I am going fast. Requirement of equipment for the microbiological section, and some of the equipment they have relaxed actually. In case the lab is not having HPLC and HPTLC or AS, so that can be subcontracted with the other laboratories. Laboratories which are already having the accreditation are approved by the drug control authorities. The services of those laboratories can be used for HPLC, HPTLC, AS. So the lab can get the approval. And for the reference documents, the lab should have the all required pharmacopoeias or other documents whichever is required for the reference of the methods. And beside that, they should have the reference samples of botanicals, reference samples of minerals, and certified reference materials of phytochemicals, reference materials of phytochemicals, and certified reference materials or reference materials of chemicals required for analysis or calibration, etc. So this was all required for the approval. If lab is having these facilities, and after inspections, the inspection authorities got satisfied, then lab can have the approval. But in the approval, actually competence of the manpower is not adjudged. Accreditation is given by the third party approval, and as per the international standard, that is the ISO IEC 17025. And for this accreditation, actually the standard which is required is ISO IEC 17025, and this is the only international standard for the laboratory accreditation. There is no other standard by which a laboratory can be accredited. So this standard is followed, and this is the history of a standard for laboratories. First standard was published in 1999. Prior to that, there were ISO guide. First ISO guide 38 was published in 1983, and then it was upgraded in as ISO guide, ISO IEC guide 45 in 1985, then in 1986, then in 99. But the first ISO IEC 17025 was released in 1999. The second edition was released in 2005, and now current edition, which has been released in 2017, is in practice. Who uses the ISO IEC 17025 standard? Laboratories who want to get accredited, they are using. Accreditation body for confirming that whether laboratory is confirming the requirement or not, and customer who want to get their samples tested, they have confidence that the whatever laboratory they have selected is accredited and competent to perform the work. And benefits of the accreditation are that it has the laboratory which has the accreditation is globally recognized and assured that it is competent and have very good quality assurance. And it also has the market advantage because laboratory has credibility, and people know that it is complying with the quality and compliances of the international standard. And reduce costs also because it may need the product to be retested and re-inspected in different countries, saving time and money. Suppose the drugs are being exported, so all over globe, whatever the report is being given by that laboratory, it is accepted. So repetition of the testing or other requirement is not there. So this is the, in essence, the international accreditation serves as an independent, globally recognized stamp of approval for a laboratory's quality and technical capability. Who can grant accreditation? Accreditation is given by the accreditation body. And what is accreditation body? Accreditation body is that organization which is accredited as per the standard of ISO IEC 17011. Because accreditation body also requires the accreditation, and that is being accredited as per the ISO IEC 17011. And accreditation body should also have approved by the regional and international accreditation organization. So APAC is the Asia Pacific Accreditation Corporation is the regional accreditation organization, whereas ILAC, International Laboratory Accreditation Corporation, is the international organization. So this, what is APAC? Asia Pacific Accreditation Corporation is the regional accreditation organization, and it has mutual recognition with the ILAC. And ILAC is the International Laboratory Accreditation Corporation. It is the APAC international organization of accreditation bodies, and it is also accredited as per the requirement of ISO IEC 17011, and it has mutual recognition arrangement with all regional accreditation bodies. This is the picture where you can see that the ILAC is the apex body, and others are the regional accreditation agencies, accreditation bodies. Sorry, EA is the European Cooperation for Accreditation. ARAC is the African Regional Accreditation Corporation. IAC is the Inter-American Accreditation Corporation. APAC is the Asia Pacific Accreditation Corporation. India comes under this APAC. SADC South African Development Community Accreditation, ARMC, Arabic Accreditation Mutual Cooperation. So these are the accreditation bodies uh, are in the operation all over the world. And in India, presently there are 4 organizations which are giving the accreditation as per ISO IEC 17025. NABL is the oldest, which is in operation since 1992. I was associated with NABL since 1993 till 2019. And QAI, it has come into existence since 2022 or 21. PADAS is also with the same period. And recently, this International Quality Accreditation Services, it has also came, I think, last 1 or 2 years. And all of them they are accredited. They are having mutual recognition arrangement with APEC as well as with ILAC. So whatever the accreditation is giving, it is at par. But NABL is very old, and it comes under the Quality Council of India. And in chemical, all the like instrumentation, chemical testing, whether the conventional or instrumental, that will come under the, but the option is there. If the private laboratory, they can have the electrical, electronics, fluid flow, forensic, mechanical, non-destructive technique, photometry, etc. also. But for the your interest is only for the biological and chemical. And how you prepare the laboratory for accreditation. So first of all, you should procure the standard. The first page of the cover page looks like this, and now the Indian version can also be obtained from the BIS, and that standards have the number ISO IEC 17025, ISO IEC and IS. Sorry, 17025 number is typing mistake on my part, but standard is 17025. And this is the structure of the scope. It has 8 chapters actually. The first is scope, second one normative references, third terms and definitions, fourth general requirements, structural requirements, resource requirements, process requirements, and management system requirements. The class 4 to 8, only 4 requirements, they are majorly having the management system requirements and the technical requirements. The laboratory should follow those requirements. And Exer A having the methodological testability, and Exer B has the management system options and bibliography, what documents this standard is based. So these are the only 4 classes which have to be required. You require the you have to follow the requirements of these whatever the requirements of these classes the lab has to follow. So first is general requirements. It has 2 subclasses, 4.1 and 4.2. It is the ethical requirement actually. So forming the foundation of the laboratory, and it has the guidelines for the maintenance of impartiality and confidentiality in laboratory. So these are very important. Otherwise, you have the ethical approach. So it is difficult to have the faith of the persons who get the testing from your laboratory. So lab has to follow the requirement of these 2 subclasses of the general requirements. After that, structural requirements. It is the structure or skeleton of the laboratory, and without this skeleton or structure, it is difficult to run the laboratory. And these are the it has the 7 subclasses. First class is 5.1 legal entity. Is the this heading is given by me, but if our class is 5.1, and it deals with the legal entity of the laboratory. Lab should have the legal entity. The second is 5.2. Lab should have a identified management. 5.3. Lab should have defined the range of laboratory activities. What type of tests they are, how many sections they want the accreditation for chemical, biological. So range of laboratory activities has to be defined. Yeah, requirement for these is given under this class. And 5.4, whatever activities lab will perform, it will follow the requirement of the management system given under the standard. 5.5 is the structure. Lab should have the defined organizational and management structure and its place in parent organization, its relationship between the management, technical and support services, and specify the responsibilities of the persons working there, authority interrelationship, and documented procedures for consistent application of laboratory active and validity of results. 5.6 is the regarding requirements about the authority and resources. So labs should give the authority and resources to the personnel who can implement, maintain, improve management system, identify the deviations if any there, and initiate preventive action and corrective actions. And reporting to lab management on performance of management system for improvement, and should ensure that effectiveness of the management system is not altered if there is any change in management system. And last is the 5.7, that is regarding communication. And this clause ensures that the communication takes place between the management system and the employees or personnel, and integrity of management system should be maintained on change of the management system. Now, sixth clause is the resource requirement. It has the six correction factor or reference value. So that should also be followed, and equipment should be prevented for unintended adjustments, so that results should not be varied. And every equipment is required to be maintained each record. So that requirement is given in the standard. Then methodological traceability. This is very important. Lab, whatever equipment lab has, they should be calibrated from the a accredited laboratory, that is that should also be accredited as for ISO 17025 requirements. And lab should also have the all required CRMs. And whenever the traceability to international standard is not possible to, then lab has to provisions how it should establish the traceability of that result. So it can be done by various means. So those have been explained in the standard. And then externally 6.6 externally provided products and services. This is also very important. Suppose you want to have the for a calibration, you are having contract with some laboratory, so that should be accredited. If you want to purchase chemicals, so that you should lay down the criteria, what type of the vendor it should be there. So these are the whatever provided external purchasing products you want to purchase. So you should have a procedure for that. This clause also require a procedure. And services, if you want to take advantage, suppose like in case that you are not having HPLC or HPLC and you want to subcontract this service from other laboratory, so you have to be assured that that laboratory should also be accredited. So then you can use the services of that. So those aspects are there in this subclass. So you require to be documented and you have to follow these resource requirements. Now next is the process requirement. Now here the laboratory, how the laboratory is functioning from the start of the sample to production of result. So again it has the 11 subclasses. 7.1 is regarding review of request, tenders and contracts. So whenever the sample laboratory receives samples, so you review whether the facilities are existing with you or not. Lab is capable. Person who will test the sample is trained. You have the equipment. You have required reference material. So all this has to be reviewed under this clause also require procedure, and it has 7 subclasses. So the lab has to follow the requirements, policy and procedures against those requirements. The second clause is regarding selection, verification, and validation of methods. You had received a sample, but under review you have also finalized this procedure will be followed. So you have selected. The procedure shall be a standard procedure, and it has been verified. And if it is non-standard procedure or modified procedure, whether it has been validated or not. So what are the procedures, what are the criteria for selection, verification and validation of methods that comes under this class? Third is the sampling. It has 3 subclasses. And sampling, suppose lab is usually lab is not involved in sampling, but some labs do the sampling also. So what procedure you have to follow for the sampling? So it is required to be documented. What plan and how to keep your samples, if any preservative is required or not, all those aspects come under the sampling. 7.4 is regarding handling of test or calibration items. So the sample which has come to the laboratory for testing, so how you have to handle. And again this class requires procedure, and it has 4 sub classes. So how to you receive, where to keep, and how it has to be decodified, and how it has to be stored or preserved after the testing, and how it has to be disposed. So all these factors or these procedures will come under the handling of test or calibration items. Calibration items mean if it is a calibration laboratory. And technical records. When you started testing of sample, then you create an analysis sheet, analysis data you get. So you have to jot down those in the on the analysis sheet. A technical record is generated, and after that you will prepare a report. That is also technical record. So all these documents, they how to maintain, how to identify, and how to retain them. So those guidelines are given under this subclass. Then evaluation of measurement uncertainty. This is also very important because you have analyzed your sample, and you whatever result you got, it is not the true value, because there are many contributions, many factors due to which result may be whatever result may be less or more. So that is called uncertainty. So uncertainty in measurements. So for accreditation, this is also one of the important criteria, that for each test lab has to evaluate measurement uncertainty and give the how much degree of freedom or result is varying from which value to which value. So lab has to calculate the measurement uncertainty for each test. That is also required in the scope of accreditation. And after that, ensuring the validity of results. When the laboratory gives the results, prior to that, laboratory uses many practices to ensure that its results are valid. What are those practices? It will use CRMs. It will use the calibrated equipments. It will use the different instruments for the same result, same analysis. It will participate in interlaboratory comparisons. It will participate in proficiency testing. It will do the retesting or repeat testing, and sometimes testing of blank samples also. To have the and record of all those is required to be retained, and procedure is also required in this case. Wherever P is taken, so the procedure is required in red letter. So this is also a very important requirement of the standard. After that, reporting of the results. It has 8 subclasses, and after analysis, you got the result, and in what format you have to report the results. So that has also been given in the standard. Since it takes a lot of time, so I have limited time, so I'm not going to. But you can have this standard from the lab. I think you will give them some document ISO 715. So you will have that document from the laboratory, so you can go. That is a very big. It requires 4 or 5 days to explain the whole document. So, but in short, I am telling you. So you have the basic format. What minimum requirement you have to mention in the report, and if you are doing sampling, so what type of the additional requirements are there. If you are reporting the conforming statement of conformity, how you have to amend the report. All those requirements are explained under this class. And then after that, complaint. Suppose you have issued the report and you got the complaint from your customer. Some mistake is there, some typing mistake maybe, or mistake in results. So lab may get the complaint also. So how to deal that complaint? The process is required to be documented by the laboratory, and you have to follow those guidelines or requirements to go through to resolve the complaint. And then non-conforming work. Sometimes non-conforming work may also occur. You just made shortcut or used some non-standard methods, so you are not getting the proper results. So those are non-conforming. Your instrument is non-calibrated and you got the not proper result. So those are the non-conforming work, and how to tackle, how to deal those. If you observe any non-conformity, so how to what action or corrective action or preventive action is to be taken. So those requirements have been explained under this class. And finally, the control of data information management. Now report has been issued and you have the data with you, and during the your from analysis sheet to report preparation, you are transferring the data and you are retaining the data with the lab. So what precautions or what policy or procedures you have to follow those have been explained in the 7.11 in control of data information management. So this was all regarding process requirements from receipt of sample to issue of the report. So this is actually the competence evaluation, because in the approval, the drug enforcement authorities, yeah, drug authority, they check if your persons are present, you have the equipments, but they do not see whether you are capable to do the proper analysis of samples or not. So this is only being adjusted during the accreditation under the process requirements. During assessment, the samples are being given to the testing personnel, and testing is witnessed by the assessors, and those samples are being given which have already been earlier tested, and what result they got during the testing. So they compare the result of the test witnessed and result of earlier result. What is the deviation? If that deviation is within the measurement uncertainty, then it's all right. Otherwise, whatever you have measured the uncertainty and you are not getting the repetition of that result, it means you are not competent or capable, or somewhere something has gone wrong. So this is the reason that your competence is being adjusted by these process requirements. So this is very important requirement, and it is actually being assessed by the assessors whenever you have the accreditation assessment. And next is this is a management system required, mainly for the documentation. And if you prepare the documents and follow those requirements of the document, it means you have a consistent system. You have not documented your management system, you will all will vary. So whole organization will have to follow these guidelines, so that there should be consistency and control. If improvement is required, so there should be continual improvement. So there are 8 sub clauses in this management system requirements. 8.1 is the options. There are 2 options actually. If the laboratory is already certified as per ISO 9001, then it can opt option B. You need not to document your management system, because in 9001 management system is already been certified. But if you are not certified as per ISO 9001, then you have to follow option A. Then lab has to document its own management system as per requirement of the standard. So what are those requirements? 8.2 is the management system documentation. You have to prepare the documents of the laboratory as per requirement. There are 4 type of the documents: level 1, level 2, level 3, level 4 documents. So lab has to document its management system document or quality manual, that is the level 1 document. The second is the management system procedures. Wherever the procedure is essentially required to be documented, lab has to document those procedures, and they are called commonly as a management system procedures. Then you have to document your SOPs, whatever procedure you are following for the testing, or work instructions to follow the to handle the equipment, etc. So third level documents having the standard operative procedures and working instructions. Beside that, you have prepared a lot of the forms and formats. Suppose analysis sheet regarding of the monitoring and regarding of the temperature, logbooks, and your um, uh, calibration, etc. So a lot of the documents are required, and for that your lab has to document the forms and formats. So those will come under the 4th level documents. And lab has to also document a policy. What will be the policy of the organization, and what will be the objective, and a policy about the declaration about the impartiality and consistent operation of the laboratory. And that document should also contain the evidence of commitment for continual improvement and effectiveness of management system, and what linkage. So referencing should be in each document. Suppose in quality manual you have given the reference of procedure, so that reference should come. In procedures you have given the reference of the formats you are using, so that should come. So linkage between the various documents should be there. and access of part of management system documents and related information for responsibilities. And all persons who are working are related with these the testing, etc. They should be given access of the management system documents. Now you have prepared the documents. Now how to control the management system documents? So you have to give a identification number to each internal or external document for their control. It belongs to the PCMH or the laboratory wherever you are working on, and ensure that documents are approved prior to issue, periodically reviewed, revisions are identified, uniquely identified, and obsolete documents are suitably identified and use is prevented. So whenever you prepare a document, so who has prepared, then higher authority should approve it, and it should be identified, given a number. And if it has been revised and the earlier version is now, it will be called as a obsolete document. So that should be separated from the running documents so that it should not be misused. So this is all about the control of management system documents. Details you can have in the standard. Then control of records. So documents, so whatever the procedures and methods you are having, standards you are having, those are the documents. And whatever is the generated during your testing, that is a record actually. So how to control those records? So you have to give a number to each and every formats, and these records should be legible, and this will be properly identified, properly stored, protected. Should have the backup if you are using the digital information, that should have the backup. And they should also have the duration time and how you have to dispose those records. So that has to be documented, and each record shall be readily available and identified, and misuse is prevented. Then control of records, yeah, you have to control these action to address, because this new document 2070 is based on the risk analysis. So this is very important. In the lab activities, you just go through where the risk can be in your working. Suppose person, he can be impartial, that is also risk. If sample is not coded, that is also a risk, because one should know from where it has come. Or relative of the top management is working in the testing, so that is also a risk. Similarly, if your equipment, if it is not calibrated, that is also a risk. So such type of the activities wherever you have any risk of in alteration of the results, so that has to be identified, and how that can be eliminated or mitigated, so that has to be documented by the lab. And this is and if suppose you identified a risk and you have taken preventive action, so that was an opportunity for the improvement. So this is closely associated. Whenever you are mitigating or eliminating any risk, so you are generating you are improving your system. So this is the reason that you have been used the risk and opportunities. Whenever you use closing a risk, then you are having the opportunity for improvement. The next is improvement. By your mitigating the risk, you are improving your system. Whatever the customers, your field formation, sending the samples to laboratory for testing. If you have any feedback from them, yeah, feedback taking is must actually. You should take feedback. If you have any information, any suggestion from them, so that is also for the improvement. And whenever you are conducting internal audit or some outside audit, and whatever non-conformity has been pointed out, and you have closed that non-conformity, that is also improvement. So this class 8.6 deals improvements. And 8.7, whenever you observe any non-conformity in your working, then you take a preventive or corrective action. So that comes under this class, and you have to make policy and procedures against the what action you have to take whenever any non-conformity is being observed during your activity, lab activity. So that is dealt under this class. Then 8.8, this is very important. Internal audit. Lab should conduct internal audit of its activities in a planned manner, either twice in a year or once in a year, and as per the requirement of this standard, mean from 4 point class 4 to class 4, covering all subclasses. Each and every class has to be covered, and the internal audit of the management system will do that fellow, yeah, that person who is not involved in the management, and internal audit of technical activities will do that person who is not involved in technical operations. So there should not be any conflict of interest. So lab has to conduct internal audit either twice or depending on okay, how much you can do, but one audit is must for each every year. So for that you should plan which month you have to do, and who will do the audit, who will do the management system audit, and who will do the laboratory lab activities audit. So one audit is required, and that audit team will prepare a report. If any non-conformity is observed, that has to be informed to the quality manager, and corrective action has to be taken within the stipulated time. So that non-conformity should be plugged in. So, and record of internal audit is required to be retained. And 8.9 is the management reviews, and every lab should also conduct one management review meeting at least once in a year. So whatever activities laboratory is performing, there is a agenda points. There are 18 aspects given in the standard. So labs should discuss each and every aspect in that management review meeting, and whatever the input has been given by the various personnel against those inputs, agenda points. So, and what is the outcome, that should be recorded, and a record of the management review is also required to be maintained. So this was all about the requirements of the. Now how you have to approach now. So lab should get it some managerial level persons for the training. Many professional organizations or these accreditation bodies, they sponsor the training programs, and the lab which is want to get accredited should get its managerial level positions for training of those training, and this training for the requirement of ISO IC 17025 standard for conduction of internal audit and management review. So at least some senior persons should get the training, and they can train to lower staff. And lab should also ensure that it is having all required facilities of the scope of testing, and it is capable to maintain environmental conditions. Lab has the competent personnel. Lab has all required equipments, and those equipments have been calibrated from accredited lab. Lab also have all CRMs, reference materials, and these reference materials and RMs have been obtained from the uh, competent authority, that is the organization which is accredited as per ISO IC 17034. That is for the that is ISO number for the reference material producer. And lab has arrangement for externally provided product and services. Lab should also ensure that quality control checks are being performed and their record is maintained. So I have already told that the use of CRMs, intermediate check of measuring equipment, replicate testing, retesting of retained items, correlation of results, review of reported results, interlaboratory comparisons, and testing of blood samples. So record of these is also required because you have to furnish details of them to accreditation body in application form. Then lab should also participate in proficiency program if it is available in the country for each group, for one test for each group of the scope. And if proficiency testing program is not available, then lab should have either participate or organize interlaboratory comparison, and the whatever result you get after the proficiency testing participation or interlaboratory comparisons, that result should be compared. Your result should be compared with other laboratory. So that is Z score. If your Z score is within 2, it means it is satisfactory. If it is more than 2, it is not satisfactory. You have some problem, and that has to be resolved. You do the root cause analysis, why you are not conforming. So now, lab should also conduct the internal audit of laboratory activities as per planned schedule. Check whether the actions against each applicable requirement of the standard has been documented and implemented. If any non-conformity is observed, then take suitable action to close that. And record of the conducted internal audit is required to be maintained. And finally, you require to conduct the one management review of the laboratory activities better after the internal activity, and you discuss each and every aspect whatever is given. There are 18 aspects given in the standard, so you discuss each and every aspect and what is the output of that. So that should be recorded. Now, yeah, this is the fourth type of the document you have to prepare: quality system management document or quality manual, quality system procedure manual, standard operating procedure and work instructions, form formats, registers and files, etc. Now, in the application, actually, these informations are required to be furnished. If you are private laboratory of a big organization, or you are like government organizations also, like PACMI, the parent organization Ministry of Ayush, and for the government organizations, no legal identity proof is required, but for the private laboratories, it is required, and it may be proprietorship firm, partnership firm, company act firm, or societies registration act firm, Indian trust registration act firm, limited liability partnership act firm. In government, actually, it is already legally identified, so need not to have the much badri. And this information is required by the in the application for proposed personnel, like authorized signatory, or like in your approval, you require to furnish who will sign your chemical test report, who will sign the botanical test report, who will sign the microbiological. Similarly, here in accreditation also, you require to furnish in this format. This is the format where which section is of the laboratory, what is your designation, what is your qualification, what is relevant experience, relevant training, and authorization is full time, part time, or which is testing. Suppose you are expert for the instrument analysis or for particular specific test or botanical testing, so whatever is your area, that is required to explain here. Then details of the staff, other working persons who are involved in testing, because their competence also be adjusted during the assessment. And third is the spoke of accreditation. This is also very important. Before, prior to apply for the accreditation, you require to prepare how many, because it is accreditation is not given to the laboratory actually. Accreditation is given to the test. You have a certificate for the scope of accreditation actually. If for how many tests the lab is competent, it has to be defined, and your competence is to be adjudged only in for those tests. So it is very important that before application, for how many, and it is not necessary that for whole laboratory, whatever your activities you are involved, even for one test you can take the accreditation. If you want, then you can take only for the pH. If you are having facility only for the loss on drying or moisture, or so the accreditation facility, accreditation bodies are so flexible. Even for one test you can have the accreditation, provided you comply all the requirements of ISO 17025. So scope is very important. For how many tests you want to be accredited. Laboratory is accredited for only those tests, not for all activities. And accreditation body is responsible for only those actually tests for which it has given accreditation. The next is major. Now you have to go, disclose the list of equipment also. What type of equipment you have, when they have been calibrated, when they were purchased, when calibrated, and calibration is valid up to which date, and next calibration is due, and it's from where you got it calibrated. This is also very important. And after that, the list of reference material. For testing, you require the reference material. So that is also you have to disclose in the application form. And details of the internal audit and management review you have to give. Detail of management review, I think I also, sorry, बैक जाने के लिए क्या करना है इसमें? अच्छा, ओके. हाँ, internal audit and management review, both you require to detail whatever proficiency testing you have participated. Those details are required to be furnished in the application. And And now you have completed your application. Now you submit, select your accreditation body. Earlier there was no choice. Only one accreditation body was there, NABL, National Accreditation Board for Testing and Calibration Laboratories, which is a board of the QCI. So now, but presently you have the choice. There are 4 accreditation body. I have already told you. Now, sorry. Submit application. Along with the application, you submit the proof of legal identity, list of authorized signatories, organization chart, list of staff, scope of testing, list of equipment, list of CRM and RM, details of conducted internal audit, details of conducted management review, details of proficiency testing and ILC participations, etc. And after that, now you have submitted your application. Now it is the action on part of accreditation body. Accreditation body reviews your application, and when it finds that it is okay, then they appoint a lead assessor, and with the consent of laboratory, they want to appoint this person to for examination of your documents and quality manual. If lab agrees, then they appoints, and those application and quality manual is being forwarded to the lead assessor. Lead assessor examines the adequacy of application and quality manual. If it is okay, then it informs to the accreditation body, and then in accreditation body appoints a assessment team, which includes lead assessor and technical assessor. Technical assessor mean who will assess the testing of the testing personnel to the laboratory, the testing part of the laboratory. So the team is being appointed by the accreditation body, and date is also being asked from the laboratory, and on a fixed date the assessment is being conducted by the team, and it is usually 2 days assessment. So lead assessor, he assesses the laboratory documentation part, quality management system, and the technical assessor assesses the how many texts are required, to depending on the how much uh, uh, bigger is your scope. Yeah, how many tests are there, how many groups are there. So depending on the your type of the groups are, how many groups, 1 or 2 or 3, depending on suppose you have taken the assessment in the chemical and microbiological and pharmacy or pharmacology. So obviously you require 4 assessors. If there are more tests for the chemical testing, you require more assessors. So the assessor will observe the technical requirements of the laboratory. Then they conduct the assessment, and after conduction of the assessment, they during assessment they give the samples which have already been tested, or fresh samples. Fresh samples are required to be tested in duplicate, or they can also give the reference CRMs for testing. So whatever result the lab gives, they compare with the earlier results or duplicate result, how much variation is there, or what result they could achieve for the CRMs, etc. They prepare a report, and report is being forwarded to the accreditation body. And during assessment, if they found any non-conformity, so that is also being pointed out to the laboratory, and laboratory is being given 1 month time to close that non-conformity. If laboratory is able to close, then it's all right. And when the non-conformity is closed, and then the assessment body put those assessment report to the accreditation committee for a review. And accreditation, it has the experts of various fields. They go through the accreditation report, and if they found that it is in order and there is no non-conformity and lab is conforming management system requirement, and their personnel are competent to function for the test whatever they have applied in the application form, then they recommend the accreditation of that particular laboratory, and then accreditation body forward the recommendation of the committee to the board, and for the approval of the accreditation. And when after approval of the accreditation of the board, the uh, laboratory laboratory is accredited, given certificate for the accreditation, and laboratory got accreditation. So this is the actually whole schematic diagram I have prepared. See how you can have the accreditation, lab applies for the accreditation, and laboratory is being examined thoroughly for the management system requirements, which covers the class 4 and 8. means general requirements covering ethical impartiality and confidentiality. And in the resource requirements, the personals are, it has adequate persons, equipments are available and they are cultivated. Lab has the proper facility, environmental conditions, measurements are traceable, and they have the external products and services as per the requirement. And beside that, the samples are also being given to the lab and their results are also being examined for their validity. And if everything is found okay, then that it means that laboratory is competent, it has proved its competence, and then accreditation is given by the accreditation body. So this was all about it. So since it has very less time and subject is very broad, only for the training of management system, internal audit and management system, it requires 3 days. So I have covered in a very short, and your session is already late and it is a lunch time. So if you have any question, then you can ask. Otherwise, thanks from my side. I have worked in this organization 47 years back. 87, I was in Ayurvedic Pharmacopoeia Committee in Ministry of Health. At that time, Ayush was not there. I was in homeopathic pharmacopoeia laboratory also in 1978. I started my career from that. And in Central Indian Pharma, I headed the R&D division of that. And from there, I was in a notified government list for the state of Bihar, Odisha, Goa, Tripura, Rajasthan, because at that time, these states were not having their own drug testing laboratory. And I had been WHO fellow also in 1992. And during that fellowship, I visited the Canada in the Bureau of Drug Research, Bureau of Biologics, Bureau of Drug, this, your, आपकी जो यूज होती हैं इंजेक्शन वगैरह क्या कहलाते हैं ये? नहीं नहीं पैरेंटल नहीं, ये मैं देखिए आई एम 70 मोर देन 72 सो ये मेमोरी के साथ थोड़ा होता है. मैं चलिए बताएंगे. हाँ, फिर इन यूके आई हैड बीन इन इंस्टीट्यूट ऑफ बायोलॉजिकल स्टैंडर्ड एंड कंट्रोल, मेडिकल डिवाइसेज एंड मेडिकल डिवाइसेज एंड रेडिएशन. In Canada, I had also been in there, Bureau of Medical Devices and Radiation. And in UK, I had been in Institute of Biological Standard and Control, National Medicine Control Agency Laboratory, and in British Pharmacopoeia Commission. And from Ministry of Finance, I had been in for the I am expert for the narcotic drug analysis also. Later I switched over to Ministry of Finance lab. These are custom central excise laboratory. 20 years I was there. So I had 16 laboratory under my control. So from there I got many trainings under the from the in USA I had been in US Drug Enforcement Administration laboratory, laboratory of FBI, laboratory of alcohol and tobacco, and from the UNDOC, United Nations Drug Office of Control, United Office of the Drug Control. So many trainings from there. So I had that type of exposure. Thank you very much. Thank you. If you have any question,
Excuse
please.